Please make a full screen.
Next slide. Yes, sir.
No, make a full
Yeah.
screen.
Yes, sir. 1 minute. दिख रहा है क्या?
Wonderful. Yes, thank you.
Yes, sir.
Good morning, good evening, all the 38 participants of this wonderful event. I saw yesterday almost I think nearly 70 were there. So I hope others will be joining shortly. I am sure that this training program ATAL Academy, sponsored by ATAL Academy, has a two component. One is the technological innovation in the Agrotech, and another, of course, is in the food processing. And I could see actually yesterday speakers were mostly adhered or were talking with reference to the Agrotech, right? My presentation more focuses on food processing because I am associated with CFTRI, and I know that 75 years what it is shown there, because CFTRI actually is celebrating now the Platinum Jubilee year. This year is the Platinum Jubilee year, 75 years, 1950s. Okay. Yeah, you can hear me, right?
Yes, sir.
Yes, sir. Yes, sir.
Yes. So, any question or clarification, you people can always ask me, and raise the hand, and then you can always clarify in between, not at all issue. Very simple topic I have taken, post-harvest technology, which is actually a very broad field, where I want to cover the two components. One is the biopreservation, and another one is the cold pasteurization. So once again, the greetings to the Swami Swarupananda Institute of Education, Bhilai. In fact, my native is somewhere in the Chandrapur, which is very close to the Pur and Durg area. So always Bhilai is our very neighboring place. In other words, can you go to the next slide?
Yes, sir.
Exactly. So I start with a primary agriculture, which you can of course see the diverse agricultural pattern in this country with a different agroclimatic zones, and now wonderful weather condition also. I hope it is equally raining in Chhattisgarh also. Now even in the Mysore it is actually quite a good rainfall, and I hope our presentation don't get interrupted in between because of this weather conditions. What we have actually enjoyed is the very good diversity in a agroclimatic zone. When I was actually, in fact, in US 2 weeks back, you will see, summer is also very intense. And I heard that the south part, like is there Indiana state is there. It is a summer. There are no snowfall. But when you go to the north, like Minnesota area is there, or Fargo is there. Heavy snowfall. And then such a actually diversity. Or you go to suppose California, not much snowfall and all these things. So such diversification, what we see in a climatic condition, definitely India actually is much more ahead. And we have actually all range of the food crops, starting with the spices. If you come to south, you will get all sorts of spices, pulses, the plantation crops, and of course the fruits and vegetables. You will get the fruits even in the cold, temperate fruits and all these things. And you see our remarkable achievement in the food production. In 2024, it is somewhere like 353 million tons. The self-sufficiency in food grain production. And then of course this target by the government is always in a higher side every year. More importantly, 2023 was a year, international year of millet, and then India actually represents a very high amount of the millet production, almost 20%. Right. So such a high amount of the agriculture productivity is there. No dearth in that. But then our problems are still persist, like malnutrition, cardiovascular diseases, diabetics, obesity, and all these things are either overnutrition or the undernutrition problems. And this is where actually the CFTRI is working for. Please go to the next one. Exactly. You see, number one in most of these things, livestock production, has the largest livestock population. The milk production similarly, right? The second largest producer for the fishery, fruit, vegetables, cereals, and how this is all this possible because we have almost climatic zones, and then different almost 15 major climates in India. The different climate, you come to south, are totally different. Northeast, it is totally different. So what this actually facilitates that actually grow a variety of food crops. You can actually look for a different, food grain production. This is how actually, diversity is, in this country. Next one. So already the 60 participants mark I can see. Okay. Why this actually the agricultural produce we could achieve, and then one of this, the reason is the green revolutions, and then green revolution everyone knows the contribution by M.S. Swaminathan with reference to India's green revolutions, right? Uh, and and as soon as you have seen the green revolution in the food crop, then you have actually the revolutions in the fisheries there. Then you have the milk production revolution is there. Then oil seed it is. So several such different green revolutions. Revolutions has happened with reference to the productivity. And one of the speaker rightly pointing out yesterday was with reference to the overall productivity. But you see, the major problem of our post-harvest losses, which is actually a major concern. You see in the cereal and pulses 30%, or in fruit and vegetables 45%, means half of the fruit vegetable goes waste, or you see I said 20%, or you see root and the tuber crops again almost 50%. You will see only less in a dairy product, though they are very perishable, but only 20%. Why? Because he knows the dairy industry, how it has been actually cooperative, been revolutionizing this country. And then the dairy or the milk production, it reaches to the consumer very efficiently after, processing and all these things. The other perishable product like fish, 30% loss, So this such a huge loss you can actually make out, almost like Australia can be fed with if we can save this much loss, right? So such a huge loss is found, which is actually the post-harvest losses, and that's why the institute like CFTRI are working on how to prevent those post-harvest losses, how the post-harvest technology can be brought in a practice, and then this raw material agriculture produce can be converted into the value-added product, can be used for the storage, can be used with the extended shelf life, So this is what actually the role of the institute like CFTRI is doing. The two basic things, one is the post-harvest, avoiding the post-harvest losses. Second is value addition to the agriculture commodity. Yes. Next one, please.
Rupali ma'am, next slide.
Yeah. Rupali madam has to be a little bit attentive because she is handling my slides. So, so she has to actually be listening continuously. Okay. So I start with the food processing. This is one of this important component of this faculty development program, right? And what is this food processing? Which to talk about. So all these are related to the post-harvest technologies. In other words, the component of the post-harvest technologies. So food processing, you will see that it is the transformation of raw ingredient by physical or chemical means into food, such as by fermentation or by sun drying or preserving with salt or addition of various type of the preservatives. And why we do the food processing? Because we basically convert unprocessed ingredient into shelf-stable, transportable food products that are desirable for the consumer and also generate a profit for the facility owner. So this is the one component. While doing the food processing, we actually aim for the value addition. What is the value addition means? Value addition represents the process taking a new commodity and changing its form to produce high quality end product. All right. Like you have the salt is there, iodized salt, or rice is there, vitamin A fortified rice, atta is there, mineral fortified atta, right? So validated product defines the addition of a time, place, or utility to a commodity in order to meet the taste preference of these consumers, right? So you are suppose just here given an example of the value addition, or the processing. You have the raw material like the wheat, and then you get a next stable product like a flour. How this flour is converted in a different range of the bakery products, right? The different range of the cookies, and then you go to Europe or US, you will get the different sides of these breads, or the biscuits. You see how this one type of the raw material is used for the preparation of the variety of the product which consumer actually can opt or is consuming, in other words. Yes. Next one. So in this direction, what safety actually is looking for, through the different translational department or the technology development department or the engineering department, we are looking for again the food processing with reference to preventing the post-harvest losses, number one. Number two is the value addition to the existing product, right? Next one. Yeah. So post-harvest technology represents or is a set of method and innovation that aims in minimizing the wastage, right? As I told that different food crops has a different type of wastage, almost 30 to 50%. How we can actually avoid those losses is the one thing. And then after minimizing the wastage, maintaining the physical and nutritional quality. We have to actually ensure the nutritional quality and produce of the produce after harvesting and enhancing the storage, because the raw product cannot be stored as such. It may get infected with this insect, or the store grain insect infested, or the get spoiled, right? So how we can actually enhance their storage or then process in a different commodity, different form, or that can actually help for the transportations, In other words, how the freshness also can be kept and then shelf life can be extended. So in other words, the post-harvest technology encompasses the methods and innovation used to maintain the quality and minimize the waste of agricultural produce from the moment of harvest until it reaches to the consumer. So what basically actually CFTRI is doing is taking any agricultural produce as a raw material and work starts from there, right? The wheat, we don't go before the pre-harvest. We look always the post-harvest one, and then taking those wheat flour and converting in a different wheat grains and converting in the different value-added product. Okay, so this is what actually the institute's mandate is. Why post-harvest technology is so important? So this is what actually I want to emphasize here, because it helps in minimizing the losses, because you can actually will be converting directly. Just like you have the tomatoes, how much the tomato actually goes waste when during the growth time or when it is produced in excess, right? So several such agriculture commodities are there when they are actually produced in a bulk or when you get the maximum yield, they actually be processed. So this is what actually the major problem is, especially in our country. Production chain, which actually can convert immediately, give the value addition and supply to the consumer. So what actually why post-harvest technology is important? Because it can maximize the quality of agricultural produce, ensure the food security. Why? Because we'll be actually ensuring in the other off-season also it is available, right? Just consider the example of tomato. If it is not processed, so it just goes a waste as a fruit. I hope it is just a 5 rupees per kg, but our onion, 10 rupees kg. But when it is off season, it goes above the 100 rupees, right? So this is where actually the post harvest technology actually plays the major role. Okay, what is the reason? It helps in enhancing the shelf life of a perishable good, because we know, just like you consider the milk, convert into the milk powder, right? And then that milk powder can be kept any years together, right? And improving the marketability, so once it is a produce process, you get the better market also. This also helps for the farmers also, because they can actually get a better income, agricultural scientists, so that they can actually develop such a variety, the food industry professionals, to ensure a fresh produce to the consumers. Now fresh produce to the consumer is very important component, which will be actually covered in by that second part of the presentation when I talk about the cold pasteurization. Okay, because any processed food, when we talk, no, it is fresh. It is the processed one, right? But when about the cold pasteurization, we actually target the fresh of the again is related to the nutritional value is retained. So this is what actually we look when we do the post-harvest technologies or post intervention of this post-harvest technology. Next one, please. Exactly. What are the causes of post-harvest losses? We actually see, you see the produce, you see the 100, but at the end it goes to the consumer, how much? Only 5%, right? So during post-harvest, steps are associated, then some material goes waste in the during transport. So this is how actually the post-harvest losses, which we were talking 30-40%, it goes like this way. Next one. Exactly. What are actually the different disciplines of the post-harvest technologies or which are the aspect which you have to actually bother much? So I will start with the microbiological point of view, which is actually the food safety, because this having a safe food, it is very much important or essential. And as Dr. Shama Madam was also told, my association with FSSI, Food Safety Standard Authority of India, I am actually representing in the antibiotics subcommittee. And how actually the food safety is playing role, because the food is manufactured somewhere and then it is transported and then sold. And then in this country, we have seen the problems of the Coca-Cola problem. We have seen the problem of Maggie, and very recently the ethylene oxide. So who is responsible for all these things, right? And then you will always see one or other problem of this diarrhea or vomiting or the gastrointestinal problem. So all these things comes. So food safety, what are the measures you take? The food safety with reference to the microbiological or chemical or the physical one. Then the next one is the fermentation, which we rely on the fermentation of the dairy product, fruits and vegetable, the sausages, the pickles, right? The quality assurance, then again, how best actually you give this quality of this product. And then of course is the processing, which is one of the important post-harvest physiology, because when you are actually packing the food or storing, suppose in a lower temperature, how we actually process undergoes with reference to the, fresh is a very important aspect, because this actually helps keeping the product the fresh, the attractive, and then it actually helps for the transportation also. Waste management is one of the important component now in the food industry, because most of this waste are the government policy with reference to the wealth from the waste, right, or the circular economy, right. So we are actually looking how the food industry waste is effectively utilized. If it is a kitchen waste also, how we can actually use that one for the cultivation of these microorganisms, and this kind of things. Or this other food industry waste, whether that can be used as a manure, or can be a poultry feed, or can be in the piggery, So such several approaches are there for the waste management also. Next one, please. Yes. So I am just giving the different methods of the food preservation in general, the green techniques, which again I will be repeating with reference to the HPP or ultrasonic or the UV or the HIS and the non-destructive technique, just for the, from the student perspective, I would say. And then these methods are actually, they are being used, the non-destructive methods are the green techniques to keep the product fresh without affecting the nutritional quality and nutritional profiling, for that purpose. And then you will see this non-destructive techniques in the previous, how they are, such as like increasing the storage life, improving the quality, micro. Dr. Rupali, can you hear me? This meetgoogle.com is there. No, you can hide that one in the screen.
Rupali ma'am, please hide that after stop sharing.
Oh, yes, wonderful. And then basically we are ensuring that the wastage is reduced. This is actually more important component, because each and everything you have seen, the large amount of food products goes waste. So how we can actually minimize those waste? Now that is the important issue in this area. Next one. Yes. So I am coming slowly with reference to the biopreservation. Before talking the biopreservation, it is important that I tells the audience what the food preservation is, right? So food preservation consists of the application of science-based knowledge through the variety of available technologies and procedures to prevent deterioration and spoilage of the food products and extend their shelf life. So why we are doing the food preservation? To extend the shelf life, assuming the consumers to must have consume product free of pathogenic microorganisms. Yeah, so you see how the microbiology actually is important in this context, or in the food industry, or in the food processing, and how actually this preservation is important. You will see that because the fruits, vegetables, and the spices, the several pathogenic microorganisms that can be spread. And that is an important concept, because the fruits are there. You don't cook and eat, right? Most of the fruits which we eat fresh, vegetables, we eat fresh, liquid fruits. We don't heat it, right? The spices, to some extent, yes, we cook and eat, but then most of so that means the microflora which is present there is alive, and that actually is a challenge for you, for your health. The culinary vegetables are there. We don't cook. We just wash it and eat, right? Raw we eat. So what does this food preservation give? Is the adequate post-harvest handling plus food processing technology and packaging technology that leads to the adequate transport and the storage. Okay. So this is what the main components of the food preservation are. So it is not only that you are post-harvest handling, but it is the processing also. It is the packaging also, and then how suitable your material is for the transportation also. Next one. Exactly. Why some food gets actually spoiled and some do not get, right? Any food microbiology, I think they thinks why the bread actually do not get spoiled, but you see the meat products get spoiled, or the fish, you can bring and keep it on the table, it will get spoiled. But then you see this bun or the flour, atta, or these fruits, they also get spoiled, but not that much, or they are resist. So in food technology, what we have so-called water activity, right? The available free water for the microorganisms or the spoilage organism to help them to grow. And then this water activity is responsible, which is actually higher in the perishable product and very low in a dried product, and that's why the food gets spoiled based on the available water or the water activity. So microorganisms, everyone knows their growth is based on the moisture or pH or oxygen or temperature, So this moisture which is present is actually responsible, and then free sugar available. Why the meat product actually or the fish products are perishable? Because of the water activity, because of the freely available sugar in these products. Next one. So once we have seen actually these methods, people have started developing the different methodologies, the thermal and the non-thermal. Then you'll see in the thermal, you have the different heating procedures, the waves, starting with the omics heating till the blanching. And then in non-thermal, you have, high pressure processing, till the oscillating magnetic field. And through this also our ozone or cold plasma, and this people are actually using for the different food commodities. And then they are pasteurizing, and this is how actually you get actually a quite safer product in the supermarket, who has prepared or packed or sterilized or handled carefully. You get much more safer product when you travel anywhere in the supermarket, right, or in the market, or in the while traveling or anywhere. I'm sure even our astronaut went to the space. Yeah. Even he also would have carried some of the food products, and definitely it has to be free from the pathogenic microorganism, right? Like this way. Next one, please. Yeah. So food processing technologies, why actually is needed? Because the microorganisms, the major concern, right? The spoilage organisms. And we are actually looking how these microorganisms can be eliminated. They can be actually controlled, or they can be suppressed, their growth can be suppressed, and then that's why we use the different methodologies, and then now much more advanced, because as the food processing is actually increased, the dependence of these people, and then shelf life which has to be guaranteed, the food has to be transported from one area into the another, right? Now most of the spices you see in the Europe or US comes from where? From India. So that has to be actually the quality process, the presence of salmonella in the meat product or in the E. coli, in the meat or this dairy product, or the listeria, there has to be a zero count. So in such cases, how this product with the assured guarantee is being prepared. So these are actually some of these methods which are actually described here, which you can see with reference to either ionizing radiations or the emerging technologies, or the high hydrostatic pressure, or the ultrasound and all these things. Next one. In what kind of food cold plasma preservation method is used? Yes, is a very important question, and people are using that one by one. Now, my actually topic is biopreservation, which I told, because mine is microbiology or the biotechnology. Then from the biotechnological perspective, what this cold plasma, there are HPPs, there are most of the engineering. The people with the food engineering, they are actually looking for some of the juice products, the liquid product, where we actually ensure that the freshness and inactivation of a microorganism fully. Yeah, there was actually the problem with reference to the network. I hope now it is all right, Shama madam. Can you hear me?
Yes, sir. We are able to hear you.
Okay, wonderful. Yes.
Uh-huh.
So I am
Second
actually
question is
talking with
also there,
reference to
sir. What is the difference in protein value of natural food and preserved food? By Srilata K. Nair. Ma'am.
yeah. So what is the difference between? Can you tell once again?
What is the difference in protein value of natural
Yes.
food and preserved food?
Yeah, so protein value will be the same, right? If you are looking for the difference or the comparison,
Okay.
and then we actually ensure that the protein, whatever this 16% is required, we actually ensure that that much proximate it comes in any food commodity, so that it is the balanced food is being supplied. So this is what actually we look for. Okay. So now let us focus. The first aspect of my presentation is the biopreservation, right?
Yes.
So everyone should be attentive, and very nice to see that there are 76 participants also to listen what the biopreservation and what exactly work we have done on the biopreservations. So my presentation actually starts from now. Okay. So I have actually defined the biopreservation here, how the shelf life can be extended rather than ionization or the cold plasma, because those are the engineering. I am talking from the biotechnology or the microbial perspective, how the microbial metabolites can be, and one of the best method of biopreservation is the fermentation, because you ferment the pickle or you make the pickle from the fruits, then you make the curd from the milk, or you make the vegetables like kimchi or sauerkraut from the vegetables, cabbage. So that is the biopreservation, and through the fermentation, right? And for this kind of fermentations, you will see the role of the lactic acid bacteria, which are actually the fermenting microorganisms. Because once lactic acid bacteria grows, because these are food grade microorganisms, when they grow, they produce a lot of lactic acid. They drop the pH, and such acidic pH, no other pathogenic microorganism will be growing. Okay, unless and otherwise it is your raw material only is initially contaminated, or your starter culture is not prepared. Okay, this is what I am trying to tell. Your milk can get spoiled, but not the curd. Of course, if it is prepared properly. Your fruit can get the spoil, but not the pickle. Of course, again the prepared properly. So this is how actually the lactic acid bacteria's role comes up, because they produces the lactic acid and several other antimicrobial substance, which I will be talking in one or other way, which I expect that participants should have more questions from now onwards, in other words. Next one. Exactly. So once we talk about the biopreservation, these are actually other important area is even viruses also can be used for controlling the spoilage or pathogenic organisms. So biopreservation here refers to the preserving the food using the biological material. Now, of course, viruses are not the biological, or are they are the parasite, but then they are also actually can be used as a biopreservatives. That is what I am trying to tell. Or as a approach of the green technology, they can be used. And I don't know in what context I am trying to tell this malware. So if any participants can tell me that what way this malware is appearing in this slide, I will be very happy. Any of the participants can give some sort of insight on that? Doesn't matter. You can put in the chat box or in the question, and then we will discuss later. Next one, please. Exactly. Now I am going to talk with reference to our research activities, because I am from the research laboratory. So now talking something out of the box is difficult for us, in other words. But on the topic, perhaps Shama madam gave me 1.5 hours may not be the sufficient, in other words. Okay. Because even yesterday also I was waiting 9 o'clock it will finish, but it was just going. And then afterwards I saw that it has to be till 9:30. And I'm very good speakers yesterday also, I could see. So I'm actually looking or going to talk with reference to the bacteriocin, which is a food grade antimicrobial agent, because if they are from the food grade organisms, as a next generation preservative. Okay. So this is what actually my talk is, in other words. Next one, please. Yeah. So this actually a molecule like no previous one. Colicin has been identified in 1928 when first penicillin was discovered, right? And colicin actually is another peptide antibiotics produced by E. coli. Okay. And after that, several organisms producing the antimicrobial peptides has been identified. And such substances or peptide produced by bacteria are called as a bacteriocin. You see here? So this scene actually comes from the colicin. Okay. So subsequently, nisin has been discovered, subtilin from Bacillus subtilis, pediocin, acidocin, saccharine, acidophilin, all these things. And then it so happened in 1988, Professor Entian, with whom actually I was there for a PhD in Frankfurt University. In 1988, this is actually Snell is his PhD student. They have identified that these bacteriocins are a gene-encoded peptide. Okay. Till then, they were considered that like a antibiotic, they are product of a multi-enzyme complexes. Like penicillin is there, streptomycin is there. But this peptide antibiotics are gene-encoded peptides, and then they have identified the primary sequences of this peptide, and then they have reported that for first time, and that's why this publication is in the Nature. Why? Because this was the first report in the history where bacteriocins has discovered that they are a gene encoded peptide. Okay, so from that angle, this publication is actually very important. Those who works in this area. Next one, and that's why I wrote the historical perspective. And then subsequently, several such bacteriocin has been identified the different organisms here. Classification: the class 1 bacteriocin is there, class 2 is there, class 3 is there, class 4 is there. And my laboratory actually has worked extensively on class 1 and class 2, which we will be discussing subsequently in the next few slides. Okay, so you will see the class 1 like the nisin is there, lactin or Mercedes is there. Class 2, which is actually we call as the antilisteria. Okay, pediocin, saccharine, leucocin, plantarocin, introcin, and all these things. Okay, next one. Now, actually, what happens is you see the primary sequences of this nisin, and then you will see no previous one. Yes, no. I have shown that there are actually typically post-translation amino acids. So this post-translation this actually thioether bridges are found. If you see the primary structure of the nisin, where serine, and threonine, they undergoes the post-translation modification, dehydration, and serine and threonine they becomes dehydroalanine, and then this dehydroalanine with this for the thioether bridge. Okay, it is not the diester bridge, but this is a thioether bridge. Okay, and this thioether bridge is being formed, in a where this serine and threonine, which actually gets modified into didehydroalanine, didehydrobutyrene, and this modified amino acids, they make the thioether bridge with the cysteine, and that's why these antibiotics are resistant to the heat. So when you add the nisin and cook the food, nisin remains active, but you take the ampicillin or penicillin or streptomycin, they gets inactivated. Okay. So whenever we in the laboratory microbiology lab, when you have to prepare sterile penicillin solution, we always sterilize by the filter sterilization. But this molecule, you can of course sterilize by filter sterilization, but you can even sterilize by heat. And why they are heat resistant? Because of this thioether bridges. And how these thioether bridges are formed, this is the mechanism. The people, those who have elucidated, identified this mechanism, as very good publication came in 1980s, those days. Next one. Now, very interesting story actually to talk about. I was saying with respect to nisin. bacterium. And then there is Bacillus subtilis. And then you see their gene organization almost similar. And that's why, Nicene and subtilin are considered as the natural relative. Why they are considered as natural relative? Because their gene organization is similar. Their primary sequence are similar. Their mode of action is similar. Okay? But they are actually produced by the two different genera or the two different group of microorganisms. So once again, to repeat, the subtilin is produced by a soil spore forming bacteria, and nicene is produced by a milk bacterium. So when Bacillus subtilis grows, you will get the new alkaline to neutral pH. But when Lactococcus lactis grows, you get the acidic pH. So beside that acidity in the medium, this antibacterial substance is being used for the biopreservation or the preservation of the cane and dairy products from 1950s. FDA approval is there for using the nicene. And why we have to actually look for this biopreservation? If there is no refrigerator, if there is no cold storage, then what you are going to do? Or if cold storage and refrigeration is there, but then there are some of these pathogens, like Listeria is there, which actually can grow in a low temperature, or the spores, which are resistant to the heat as well as the low temperature. They will be probably like Clostridium spores are there. So how you are going to actually control or ensure the food is safe from such a pathogen? I hope it is quite convincing to everyone. Why biopreservation using this molecule is required? That is what actually I am emphasizing. And from that angle, you see this diversity which has been people have been studied. Next one. Okay, so I will explain with reference to one of our first molecule which you have actually discovered is the enterocin from the Enterococcus faecium. And what we have identified is that like the nicene, which is actually produced by a quorum sensing mechanism, you will see that even enterocin actually has the similar mechanism of the production. And how you see here the zone of inhibition around these colonies, when they are actually induced, the bigger zone of inhibition is there. And why that bigger zone of inhibition? Because some inducer molecule was added. And what is this inducer molecule? In the culture filtrate, you will see some sort of a peptide which is responsible for its synthesis. Okay, like in case of nicene itself trigger the expression of its own, right? But here it is a factor which has been characterized in our lab. Next one. Yeah, now I am actually telling the another example. First one I told about how the inducible enterocin is being produced. Now I am talking with reference to the enterocin produced by Enterococcus faecium. And hence now what happens is the most of this antimicrobial substance produced by a gram-positive bacteria, they kill only a gram-positive. But then we have important are the experimental evidence that even gram-negative like E. coli also can get by well diffusion assay against Listeria here and against E. coli, and then we could see that the crude culture filtrate of Enterococcus faecium could inhibit the E. coli. A surprise, totally new information. It won't happen. Not many such informations are available. And we did a partially purified the substance, separated down agar or gel electrophoresis, and avoided that when the pathogen. You see here E. coli is getting inhibited. Okay, go to the next one. Then what we did is we took this E. coli culture and added this partially purified antimicrobial substance, and did the scanning electron microscopy. Next one. Exactly. Okay. So you see here, so after 4 hours of treatment, most of the E. coli got ruptured and was inactivated. and this work actually is there in this publication almost now 13 years, which we have discovered that the Enterococcus faecium, antimicrobial substance from Enterococcus faecium, has the ability to kill the gram-negative pathogen. We have such evidences. We have the publications. This is actually the finding from our lab, and whoever tried to question us or sought the clarification. Then we have to tell that it is because of the lipase in combination with the bacteriocin was a responsible molecule to make the pores to the E. coli and inhibit or make the cell lysis. So this is what actually the explanations which we have given in these publications. Okay, so after 2 examples about the Enterococcus, now I will go to the next one. Next slide. Yeah, we actually identified several bacillus culture also. Okay. For the antimicrobial substances, and then we had the methodology to study their antimicrobial substances. Why these antimicrobial substances? Because these antimicrobial substances directly can be brought as a food preservatives. Why for a food preservatives? Because they are a part of the biopreservation, right? So we can actually see the different bacillus culture which we isolated from different fermented food. They had the molecular weight of varying sizes. You see here. This is little slightly bigger, this is little bit smaller, this is much smaller. This such a varying are the antimicrobial substances representing the high diversity we have identified and characterized. Next one. Yeah, here actually we changed our methodology of a biopreservation. What we did is that antimicrobial substance from the bacillus culture, we actually incorporated as the LDP, the packaging film itself we have soaked with this antimicrobial substance preparation, and in another case we sprayed, and then you can actually see, the pathogens like Listeria, Staphylococcus, or Bacillus, or Micrococcus luteus was inhibited. That means this LDP film was had an antibacterial property, and this antibacterial property comes from here. The antimicrobial substance produced by the bacillus culture, when it was incorporated or soaked in the solution, became antimicrobial. And such packaging film you use, your food remains the same. Your food product actually remains the same from the pathogen. So that is what the purpose is. Or the here our hypothesis was. Next one. Yeah. Now I am going with reference to the 4th example, and this is actually from the pediocin. And I told that pediocin is a class 2 anti-Listeria bacteriocins, heat-stable molecule, and exclusively works against the Listeria. What we actually identified is the different pediocin producing bacteria we identified. What happens in case of the pediocin is that pediocin actually is produced by pediococcus, right? But we have actually the evidences that pediocin can produce by lactobacillus, which is reported, but enterococcus is a new evidence from our laboratory. There was a question with relation to nisin and subtilin, and I told that nisin is produced by the milk bacteria, subtilin is produced by the soil. Nisin also inhibits the several gram-positive pathogen, including the spores. Subtilin also on the similar way. Okay, both. Nisin is commercially used, bacillus is subtilin is used relatively less. And related to subtilin, I have 2 more again examples. There I will cover. I hope it is okay. Is it okay, Shama madam? There was a question on the chat box about the nisin and subtilin activity.
Yes, sir.
Yeah. So I told already, and then nisin and subtilin story is not yet over. I will be explaining about our own findings in a few minutes from now. Just a minute. Participants can be actually posting their questions so that I also remains alert and then can make out that where I am actually missing. Okay, not at all issue. I could say 81 participants have joined. It's very good number. And I hope everyone is actually paying attention for the presentation.
So 91 have joined.
Oh, wonderful. So, and I hope most of them are from the biology or biotechnology background, so they will not have much difficulty, except maybe the Rupali madam, perhaps. Because I believe, you
You
know,
are from computer stream also, sir.
exactly. And that's why I have shown that a malware virus, that we are also related with some virus only, but it is little bit different. Okay. Now I am talking with reference to the pediatrician, and very important studies actually emerged from our laboratory. What I am trying to tell you is that pediatrician production intergeneric and interspecific has been reported. But we have discovered within the interspecific is especially intergeneric from enterococcus. Okay. So so we actually isolated different cultures producing the pediocins, seen by the west dot plot, like the southern plot. Seen their microscopy, you can very easily see pediococcus means is a cocci. These are cocci, but this is not a cocci, right? This is rod, so it is a lactobacillus, right? And then we have seen actually their plasmid profiling, and then these things are, southern hybridization. And next one. Yeah. So this actually we have confirmed, and then you can actually see that pediocin actually is produced or reported initially by pediococcus acidilactis. But you see the result from our laboratory: streptococcus, enterococcus, lactobacillus plantarum. So these are all our results from our lab, and that's why we call it that this is the intergeneric and interspecific pediocin productions. Which was totally new information. All this culture is actually kept in -80 and safe. Who will use? I also don't know. In other words, so you will see the related publications of this work. But we have otherwise not only with us, but these cultures are actually with the Culture Collection Center also. You see, NCIM Pune, NCL Pune, all this culture. Here is the number. So this number actually is of the culture collection number, but these cultures are isolated, identified in my laboratory by my PhD students, and the study has been published. Okay, next one. Yeah, we actually try to look how this gene organization is there in this. So first one you see, this is actually the standard culture or the reference one. Okay, pediococcus acidilactis CH. Those who work on pediocin, they will know. Okay, this publications from the Bibekre or from this Bennick from Netherlands. Okay, but you see, the information from our lab is here: enterococcus study, and then this lactobacillus plantarum. These are our cultures, and then you see almost similar gene organization has been found. Okay, so what people have reported previously in a pediococcus acidilactis, almost similar gene organization has been reported by us. And this is actually this evidence. Why we are actually telling that our work is a novel and new? Because we have such a scientific evidences to claim those results. We did first and told the scientific community that such information exists or such a new discovery is there from our side. So this is what actually the information is. So genetic, molecular evidences, the biochemical evidences, microbiological evidences helped us to make those new claims. Next one. Yeah. So when here actually it is produced by the different organisms, what does it mean? So that means if it is a lactobacillus plantarum, it grows in a different conditions. It requires a different salt concentration, right? Then it requires the different pH. Enterococcus, totally different conditions. So you see, we actually checked the antimicrobial substance or pediocin production in different temperature, different pH, and then different salt concentration. Like suppose if you are doing a pickling, you add a high amount of salt, right? So in such salt concentration, you can actually get this antimicrobial substance production, because some of this pathogenic bacteria, they can actually grow in presence of salt also, or listeria. No, so how you are going to take care? And the pickle, you don't cook and eat, right? You eat whatever it is prepared. That means if it is a contaminated, you will be eating the live organism also. But if that your pickle is fermented using this antimicrobial substance, that means your pickle is safe, secured, and that's why Sometimes the chutney is there, pickle is there, even they actually causes the foodborne diseases. The reason is because of the unsafe production, because of the presence of some pathogenic bacteria, because of not using a defined starter culture. These are the reasons. And then when you are actually going to make this product in the industry, in the large scale, then you can see what is the fate. Since we are making this most of this product at a household level and consuming, in a small community, so we don't much bother with reference to food safety. But if it is produced in a larger scale, when it is produced in one place and then transported to the other place, then food safety is really the issue, right? I missed that question actually. I don't know how to see that, but I can. To what extent use the bacteriocin justified as these are inhibitive only to the related family? Exactly. So we actually look for the specific pathogen or the target organisms like Nisin is there, there is a Clostridium spores, right? Or the Pediocin is there, again the Listeria monocytogenes, and Listeria you will see vegetables, dairy products and all. And then like a antibiotics, these bacteriocin, they don't kill all the organisms, they kill only the closely related and then pathogens. But then you are actually using the food grade material to protect or preserve your food, to extend the shelf life. You are actually ensuring that such pathogenic organism is, your food is free from such a pathogenic organism. That is what actually I can answer. Okay. Next one, please. Exactly. So this actually intergeneric and interspecific, what we try to find out is that initially the master culture of Pediocin is a Pediococcus, which actually we have drawn here. Okay. And from here, perhaps, just like how human civilization has happened, and from Africa, people came to Europe or Asia, and from there only they moved to Asia, like this way. These genes would have migrated. Like that way, we are actually given this hypothesis. And this paper actually is there in the Journal of Basic Microbiology, I believe. Okay. Because what we have actually seen, our people are also have reported that there are actually the transposons in the flanking region of this operons or the gene cluster. And those flanking regions are responsible for moving these genes from one organism to another, from one species to another one, from one genus into the another one, okay, within the bacterial community. So this is what actually reason we try to give with relation to the Pediocin. Why? Because with reference to the Pediocin, we had a totally new findings, informations to report. Okay. Next one. After Pediocin, I am actually talking with reference to the plantarocin, and then plantarocin also we actually investigated because plantarocin diversity is actually very high. Okay. And then Lactobacillus plantarum itself, we have actually a database itself, the Lactobacillus. Yeah. Rupali madam can immediately check in the internet and see what is this Lactobacillus. So Lactobacillus is the database exclusively meant for the food grade Lactobacillus cultures, which actually are used as a probiotics, used as a starter cultures, for food applications, and has several health benefits. One of the health benefit actually comes because they have the ability to produce the antimicrobial substances, and this antimicrobial substance is referred as a plantarocin, and we actually characterize these plantarocins, and our actually culture, this is actually the our cultures here, E1 culture, okay, has a totally different type of gene cluster, okay, gene organization. You see this molecule with this much space here, it is available, was found in our studies. Okay, next one. And then this Lactobacillus plantarum cultures, we actually explored for their probiotics application because they had the exceptional ability to adhere to the intestinal cell wall. Okay, go few spot first with relation to this slide, because this is actually very important and interesting slides for this all the participants perhaps. So two more, the upper one. Can you go back a little bit for the same slide? No, next one. Just a one enter.
This one, sir? This one, sir? This one, sir?
Next one. Yeah, I am now talking with reference to the biosensor, because we are actually using the cellular biosensor for detecting the antimicrobial substance for their mode of action. Where this antimicrobial substance actually inhibits in a pathogen or the sensitive culture. For this, actually, we use this cellular biosensor, which are actually genetically engineered, and then principle actually is very simple, which is actually explained here, that the outside stress or the antimicrobial substance or the inducer molecule actually activate this histidine, being recognized by this histidine kinase, and this histidine kinase, phosphorylate the response regulator. This phosphorylated response regulator activate the corresponding gene, promoter of that gene, and then this promoter is fused with the beta galactosidase. So whenever the extracellular substance is present in a stresses in a nanomolar concentration, is enough for inducing the beta galactosidase, LAGZ gene. Okay, and this expression actually helps you to identify the mode of action of that antimicrobial substance. So when we say any antimicrobial substance, it will have one of the antimicrobial mode of action, one of this mode of one of the it will have one of this mode of action, like it will inhibit the cell wall biosynthesis or protein or DNA or RNA or the fatty acid, right? So this method actually rapidly helps to identify what is the mode of action of a given antimicrobial substance without purification or without producing in a more amount in a in a high throughput manner. What is the meaning of high throughput? At a time, you can actually analyze large number of samples, just like in the microtiter plate. So here is actually the schematic representation, how this biosensor or these 5 different biosensor which are there in our lab, they actually are being used. Okay, can you just press once? Exactly. So this yes, so just a minute. Okay, so we have actually the YPU for the cell wall, YHE for the protein biosynthesis, then the for the DNA biosynthesis replication, the YOR, then for the RNA we have the YBGS, and then the fatty acid we have the FABH1, and all these biosensors are exclusively tested, no previous one, with their reference substance, like the cell wall was tested with the penicillin, the protein one was tested by the neomycin, the DNA was tested by the nalidixic acid, the RNA biosynthesis inhibitor biosensor was tested by the rifamycin, and then fatty acid, the triclosan. Okay, so such reference substances has been standardized. So you give me any antimicrobial substance, within the next day, I will be able to tell what is the mode of action of that antimicrobial substance. If you have to do classical methodology, it may take months to get it.
Tiny ma'am, please.
Yes. Participants are paying lot of
Please
interest.
continue. Please continue. All
Yes.
of the participants, please mute your mic from your end.
So what I am trying to tell you is that these cellular biosensors are genetically engineered bacteria developed in our laboratory are being used for studying the mode of action of any antimicrobial substance. So suppose if you even give me ethanol, which is or SDS, which is not labeled, and then you ask me what is the mode of action. So in such case, I will use all this 5 and I will see the blue coloration for the cell wall. You give me suppose the chloramphenicol, which is the protein biosynthesis inhibitor, and you don't label or tell me, only you tell test sample. It will not give blue coloration anywhere, but it will give for the protein biosynthesis, Or you give me suppose the rifampicin, which is antibiotics, is used for the treating mycobacterium. Okay. And where I will get the blue coloration, I will get the blue coloration only for the YVGS reporter and not anywhere. So what does it actually tells? No, it will actually helps me to classify or identify that given antimicrobial substance rapidly and go to the next step. So that is what actually the purpose is. So how did we use this one? That story and this thing is all right, but did we use? So yes. So answer is in the next slide. Yeah. So we use actually to characterize a antimicrobial substance produced by Bacillus licheniformis. Okay. And this Bacillus licheniformis was showing giving reaction with the subtilin biosensor. Okay. First the cell wall and then subsequently the subtilin. And then we actually were interested because subtilin is produced by Bacillus subtilis, not by Bacillus licheniformis. Okay. And this is actually the report from our lab that we actually told first time that subtilin can be produced by other species than Bacillus subtilis. Okay. So these are actually the experimental evidences for that. Next one. Yes. Yeah. So all these are actually explained here, you see. And you see, this is the publication from there. So this is actually the second or a third paper from my side with the single author. Shama madam was praising me, always sir will be busy in one or other things in the research and activities because I will not keep quiet. So you see this one, that interesting paper also is with the single author. This is the second paper with the single author, the sublichinin. Yes, what is the sublichinin? New subtilin-like antibiotics by Bacillus licheniformis. Okay. And what is the, how actually this was carried out or proved? By using that biosensor methods, by using this LCMS method, MALDIT of MS, and then heterologous experiment, and several other experimental evidences, in other words. Next one, please. Yeah. Now I am actually coming for this, that what I was talking about, the subtilin or subtilin-like. This is about the nisin. Nisin actually production we have actually reported, of course, from the lactococcus, but we have seen upon sequencing different variants like nisin cube. Okay. In case of nisin, there are actually 10 variants are identified. In our study, we also identified few variants, especially the nisin cube. Okay. And for this also we use the nisin biosensor. And you see the nisin beta-galactosidase induction, which I was talking about, the lacZ gene expression is given here. That different our lactocose lactase also shows the different level of this induction. So this is what actually the nisin story is. Next one. Yeah, that one. This is about the subtilin. You see again the subtilin, and then this is actually the Bacillus subtilis, but this subtilin is not like a subtilin, it is like the sublichinin. Okay, that picture actually is not there, or maybe in the next slide if it is at all. Okay, so this is what actually such variability, such a diversity, such a potent molecules, their antimicrobial activities has been reported in our group, which we use partly for the biopreservation purpose, which I have not shared those results here because I thought better I actually tells how we discovered these molecules and concluded that they are really new. They are the novel molecules identified in our studies. Next one. So with this, the first part of my presentation I am concluding here. These are actually few of these people. So the presentation is not yet over. Now the second part is going to start. Okay. Okay. I think this some more information is there. Can you just make that slide bigger? Very interesting. Actually, information is here in this slide. Slide number 39, I guess. If participants has any questions, they can always ask me. I will be very happy. Now already 7:80.
That to what extent, Mamta Sharma ma'am has asked, to what extent is the use of these bacteriocins justified, as these are inhibitory only to related family members?
Yeah, I answered this question previously, as I told, related to the nisin application in the cane and dairy products for the Clostridium, right? or the vegetable and the dairy product preservation, the pediocin, limited is there, but then still. But while doing so, you see actually very important information is given here. Previous one minute. Yes. We actually discovered a RNA inhibiting antimicrobial peptide with the similar activity of rifampicin. Okay. And then this molecule is under study right now from the two different probiotic bacteria. We call them as a lasopeptide. 3 students are working presently on a lasopeptide. And how did we identify it? By using these cellular biosensors. And what is their application? Treating the mycobacterium. Otherwise, you have only the rifampicin, and then mycobacterium, how the excessive drug resistance is. And this molecule actually they don't have the problem of the drug resistance. Okay. Besides that, this I think I can't tell or can't talk because our application is different right now. Okay. Okay. So this is what actually the story was with reference to the bacitracin, the subtilin-like, and then RNA inhibiting molecule also. Next one. Yeah, I presented already. I believe, I don't know, somewhere it may be duplicated this slide. Next. Yeah, I was talking with reference to another Bacillus subtilis, which is actually producing the subtilin-like, but it is actually sublicanin. And this actually produces one more antimicrobial substance. That's why we call it dual bactericin, the subtilocin. And due to this reason, because Bacillus bacteriosis of the gram-positive bacteria, they will kill only the gram-positive, not the gram-negative. But you see here, Klebsiella, Salmonella, they are gram-negative, right? And we get the zone of inhibition, And what is the reason? Because this culture produces a subtilocin, and this synergistic action perhaps is responsible. When I was talking with some of these companies, they were very much interested with this molecule, but they want to be produced in a fermenter in a large scale. Okay, and this is what actually the limitation is, but still we are actually looking for. Okay, so I hope one or other day all these our molecules or cultures will be of importance, and then anyone will be asking that yes, the work which was presented in this faculty development program was the basis of of this history and then the scientific studies, if at all, if it emerges. You see the Salmonella or the Shigella or Klebsiella, these all are human pathogens, and then we have the substance or the antibiotics to kill this. We have the safe substance. We have the substance which do not have the problem of a drug resistance, right? To kill this pathogen. Next one. So here is actually the answer to the Mamta madam's question, perhaps, how the common bacteriogens and their usage has been demonstrated. You see in the dairy industry, meat industry, and vegetable products, right? We of course use the very minimum, but then you see, the abroad or Europe, US, they have to actually depends on the cold storage, and in the cold storage also some of these pathogens they grow. Then they have to actually depend on the processed food. They have to depends on the food prepared, these food products prepared, months together, So in such cases, what, we always take the vegetable, the freshly grown one. But this is not the case. You go to US, Europe, you have to purchase from the supermarket, right? You will not get our sabji mandi there. You go and get the vegetables, E. coli problem was there in lettuce in Germany a few years back, right? And then this vegetables, leafy vegetables, particularly we eat raw, So that means we will be actually consuming this kind of pathogen also, if they are actually not checked properly or processed properly or studied properly, in other words. Next one. Yeah, there is actually the legislation with reference to nisin, and the nisin actually is permitted even in India. It is being used. Recently, there was actually netamycin, netamycin actually, which actually inhibit the fungi, their spores, has been actually introduced, the peptide antibiotic netamycin, in AFSS actually has permitted to use the netamycin in the dairy industry for the preventing the fungal growth, fungal contaminations. And then it knows that a fungi actually can grow in a refrigerated condition also, right? Then how you are going to control that? So one of such molecule, in other words. Next one. Yeah, we have actually developed this another shelf-stable probiotic curd, which I will not explain much, but what I want to try to do is that this product actually you can keep some 15-20 days also in the room temperature, and then it remains. And then we actually check that this product itself, you spot it, it has actually antimicrobial activity. Now we are actually in a process to develop this one further, that full technology, and then see if this can be actually commercialized. Next one. Yeah, very interesting actually study by one of the students. She just submitted the thesis three months back. We actually use this lasopeptide, which is referred here LP2514 in the preservation of the meat, chicken meat. And then you see several pathogens. When this LP2514 was used, the pathogen was really reduced. The Listeria monocytogen, the Bacillus, even the Salmonella, except the coliforms, or even the Xerops also, you see, very clear results, And when we actually applied this one, and then see, you can actually see the freshness of this meat product is kept longer. We know chicken, these things we always freshly cut use, but then sometimes you never know how many days it is. Means not days, hours perhaps. And then what is the shelf life of that, or what type of the pathogenic or spoilage microorganisms are there? No one is bothering, I think, nowadays. But if you have to actually keep it in a refrigerated storage conditions, or the store little bit longer perhaps, then what is actually the measures? And then some of this pathogen actually can grow in a refrigerated condition also, as I told. So what you will do in such cases, right? So this is how actually the molecules, and this is a study from our lab, where we have actually demonstrated the preservation of chicken meat, and very clear results, okay, appeared from our laboratory. Next one. Exactly. So this is actually just conclude the first part of my presentation. It doesn't mean that again you have to wait for a 1.5 hour. It will be only some half an hour only. The next part, and that is about the cold pasteurization, right? So Dr. Rupa Verma is asking what media could be used if you want to. I missed the question.
Sir, how large scale production and screening of the bacteriocin could be done by using other bacteria which is not known yet?
Exactly. Very interesting. Very interesting.
And the other question by Dr. Rupa Verma is that what media could be used if I want to isolate from other bacteria? Yeah,
so it depends on the selective media, or the media which is required for that bacteria. Like if it is a lactobacillus, MRS, bacillus, nutrient agar, right? So accordingly, you can actually use those bacteria, those media, and then your bacillus, if it is there, then we do the heat treatment so that the vegetative other organisms can be eliminated easily. Only the spore forming bacillus is there, and then you plate it. Okay. But if it is a lactic acid bacteria, MRS, bifidobacteria, MRS with cysteine hydrochloride, and all these things. I hope it is quite clear. Yes. So next one. Yeah, so we are actually concluding the first part of the presentation. Now I am going for the cold pasteurization. Okay. And then once we say the cold pasteurization, it is actually the pasture or the pasteurization is the process, and then you can actually see the procedure that heating the milk, high temperature, shorter time, or the medium temperature, longer time is the process. And then what actually happens is that the enzymes, in the organism presents, they get denatured, and then since we rapidly cool it, that enzymes remain separated. They don't come in the normal case, and that's why the pasteurization is effective. It so happened actually 2023. Yeah, no, last year only. I was actually in the Paris, and when we know that Pasteur Institute is there in Paris, so I could actually go there. So this, if you see here somewhere, Pasteur Institute, this is actually in the Pasteur Institute. I am standing, at least for the photography purpose. Of course, they didn't allow me to go inside. Not like India, that foreigner comes and then you get a very good treatment. But then I thought, no, I should visit the Pasteur Institute, at least that area, you know. So this both the photograph actually from the Pasteur Institute in the Paris, you know, from there is. So everyone knows how the pasteurization, I think we would have not had the safe milk early morning if there would have not been the pasteurization process discovered, right? You know, now we knows that the milk which we get from the dairy next morning is free from the disease causing bacteria like yellow fever. It is a safe, we knows that, you know, it is actually processed properly, we knows, you know. So all this actually contributes, goes to the Louis Pasteur, who has actually developed or demonstrated that the live organisms are the cause for the spoilage, you know, when he was studying this alcohol fermentations. And this is how actually the pasteurizations process came and is very effective in the for the milk. So I'm not talking about the pasteurization, I am going to talk about the cold pasteurization. Next one, please. Yeah, so what is actually cold pasteurization is? Okay, so we are actually not heating, but we are actually sterilizing the food system, you know, by you are irradiating and ensuring that the harmful microorganisms are eliminated without heating. Exactly. There is a pasteurization and simple pasteurization. This is what I am telling. Now I told first the pasteurization. Now I am taking cold pasteurization. That is the answer to your question. You know, if I would have told directly cold pasteurization, then somebody would have told what is the pasteurization. Pasteurization means who has discovered. You know, did you see the Pasteur Institute's answer is yes. That's why I put my photograph also. That I visited Pasteur Institute in Paris. I hope it is clear. What do you say, Chama madam?
Yes, sir.
Yeah, exactly. Okay. So this is what actually the cold pasteurizations, that eliminating the harmful microorganisms without heating. Okay.
Yes.
This is the cold pasteurizations. Okay. And why actually it is important? Because the taste, the flavor or texture of the food commodity, you want to keep it as such, as a fresh. Okay. The fruit juice, you don't want to heat and drink, right?
Yes.
But you want to drink without the harmful microorganisms. So you have to do the cold pasteurization. Okay. And that's why you go to any restaurants now, most youngsters, they will be drinking like this way. Little bit slice lemon and then different type of juices and all these things. Yes. Since you drink occasionally or when you are health are prepared properly, it is all right. But then, you know, suppose if it is not hygienically prepared, then you never knows what will happen. Okay. Next one. Yeah. So yes, in the cold pasteurization, how we do is high pressure processing, because you are not heating, pulse field electric. These are all are the engineering principle. Please don't ask me any more difficult question now related to this topic. These are actually the different methods being used as a common cold pasteurization. Okay, cold plasma. Don't ask which how cold plasma works and how did you do. Okay, I did a cold pasteurization. I will tell my example subsequently, or ultrasound, ultraviolet, or ozone treatment.
Okay.
All these are actually segments are different approaches are examples of the cold pasteurization. Next one. Yes. I am actually using a chemical substance for a cold pasteurization is hydrogen peroxide. Okay. And what does the hydrogen peroxide does? It actually causes this oxidative damage to the microorganisms. Okay. And because H2O2 directly reacts, generate this oxidative species like superoxide oxygen and hydroxyl radicals, and this one actually acts, damages the DNA, the protein, and results into the cell death. Okay, this is how actually the hydrogen peroxide is used. Okay, so what are the advantages of using the hydrogen peroxide? It is given in the left side. You will see decomposes the water, less toxic, and more important, it is a food grade. Okay, so when it is a food grade, that means you can use for the food preservation. Okay, so my actually presentation of the second topic starts from this slide. But there are some disadvantages which are described here in the right sides, which I will not read much at this moment. Next one. Yeah, you see the hydrogen peroxide in food industry, for what are the purposes it has been used? For the sanitization, for the disinfection, for the packaging, disinfection of packaging material, environmental, disinfection of the environmental related food industry, sanitizer, disinfection of the water, disinfection of some of this food also. Several such applications are there. Okay. Next one. Yeah. Now for what purpose? No, previous one. What purpose I use the cold pasteurization? I use cold pasteurization for sterilizing the milk. Otherwise, what is the method? You boil it, right? Or keep it in the refrigerator, So what we did is we use hydrogen cold pasteurization using hydrogen peroxide for milk sterilization. Okay. Now the milk actually sterilized by the heating, but in that case, what happens? Nutritional value and flavor of the milk will be altered. Okay. So when you don't want the quality of the product to be altered, you use this cold pasteurization methods. Okay. And that's why this cold pasteurization hydrogen peroxide is used to kill the microorganisms. And how this hydrogen peroxide actually works is described here, as I already told, that reactive oxygen species or the causes the oxidative stress and damages the nucleic acid as well as the protein molecules. Next one. Yeah. What happens actually after treating with the hydrogen peroxide? Actually, this slide came before. Maybe we can go to the next slide and then come back to here again. Okay. Next one. Yeah, we use hydrogen peroxide, you see 0.05% hydrogen peroxide incubated at 45 degrees, because at that temperature maximum reaction will be there of hydrogen peroxide. And then whatever the residual hydrogen peroxide, because you can hydrogen peroxide, how it is actually harmful to the organism, it can be harmful to us also. Okay. Or the microflora present in our gastrointestinal tract can be harmful to that. We use the catalase enzyme. What does the catalase do? Catalase breaks down the hydrogen peroxide into water and oxygen. Okay. And then we actually sterilized we treated the milk using hydrogen peroxide. Means we did the cold pasteurization. And why, for what purpose we had the catalase? To remove that residual hydrogen peroxide present. So this is what, to do that, we isolated the catalase producing yeast culture. So this is what actually described in the previous slide. Can you go once again to the previous slide? So we isolated the yeast culture, which is actually producing the catalase, and then this catalase is used where cold pasteurization was carried out using the hydrogen peroxide. And then you can actually see the 72 hours of incubation, maximum catalyst production and as well as the biomass was. So this we actually use this time scale for the maximum biomass and then catalyst production. And then this enzyme produced from the yeast Saccharomyces cerevisiae was used for removing the residual hydrogen peroxide that was used for the cold pasteurization. So now you can jump to the next slide. Next slide, please. Yeah, exactly. So I already told. So how actually you see here, in a 0.5 hour, 96%, whatever was the total residual was there, 96% was removed. But within a 1 hour, the most of this hydrogen removal of this hydrogen peroxide we have reported. Okay. So this is with relation to this is with relation to the removal of the hydrogen peroxide. And then we have actually seen the whatever the initial count was there of almost 3 log, it was actually reduced to the 0 log upon the treatment. Okay. And then that milk was sterilized by the hydrogen peroxide, and then residual hydrogen peroxide was removed by using the catalase, which actually we have developed that enzyme or the culture producing that enzyme was studied in our laboratory. Next one. Yes. So to conclude this part of this presentation, we use actually this catalyst for the removal of the residual hydrogen peroxide, which was used for a cold pasteurization. Okay. So this is what actually the overall aim was. So what this actually we conclude from here, that we have isolated a potent catalyst producing organism, yeast Saccharomyces cerevisiae, and then this that was the first part. Another, the enzyme produced was very effective for removing the residual hydrogen peroxide from the cold pasteurized milk, wherein almost 99%, whatever was left over, most of the hydrogen peroxide we could remove it. And through this method, what we are actually trying to tell is that without altering the texture or the flavor of the milk, you can actually do the cold pasteurization using the hydrogen peroxide or the cold pasteurization, and then residual, that substance which is present, can be effectively removed by the hydrogen peroxide. Okay. Next one. So with this, I am completing my presentation. I think here it is only with relation to the application in the food industry, how the different cold pasteurization methods works in the dairy, the meat product or the fresh produce and all these things. Most of these are the engineering methods which I have already was enlisted to you people, are ready to eat meats. Okay. What are their advantages of this cold pasteurization is described in the right side, especially with reference to the preservation of nutritional value, because these are non-thermal technique. So you are actually these are actually avoid the nutrient degradations associated with the heat, maintaining the level of vitamins, because the vitamins actually gets degraded when you are actually heating, right, or cooking, or some of this antioxidant actually gets are being preserved through this cold pasteurization. More importantly, the sensory quality, the color, flavor, and texture is also remains unaltered. Okay. The shelf life extension, by doing all these things, cold pasteurization, you can actually reduce the microbial load substantially from this food. And more importantly, what we are actually aiming at the end is the food safety assurance, right? So effective in reducing the pathogens such as Listeria, Salmonella, and E. coli, because hydrogen peroxide will not discriminate whether it is a gram positive or gram negative. Maybe a little bit dosage is only the issue, but otherwise it will kill all these things. And then we will be basically minimizing this chemical preservatives, which actually is the major cause, right? Some of this chemical preservatives, if you add the salt also, is actually the problem, of this blood pressure, right? Then benzoids, sodium benzoate actually also is a carcinogen if it is in a,
Yes, sir.
right? So several such actually conventional preservatives, chemical preservatives, can be minimized by this method. But then there is actually need for you people to do this research and then see how best it can be used, right? The last one is this actually enables production of clean label functional food that meets the modern dietary trends, because people wants to eat fresh, So fresh means you have to go only the sabji mandi or in the farm. And then now everyone wants to be dependent on the supermarket. And then how we will get otherwise in the supermarket? So these are actually the methods which are actually being developed by the food technologists by using the post-harvest technologies, method interventions. And then they are supplying to us the fresh one, right? More fresh than the Reliance fresh also, I can say. In other words, exactly. Shama madam, Shama
Few
madam,
questions are there, sir.
yes, smiling. That means she has understood what I want.
Yes.
Next
Few
to
questions are there.
just I think conclusion slide is there. I
Okay.
hope it will be very difficult to view. Time is only 7:45, so yes, it's officially I have 15
Sir,
minutes.
it's exceeding, sir. Your time was up to 7:30 only.
Oh, is
But we
it?
can take a few questions. Shanti ma'am has asked that.
7. Oh, yes. I thought it is 8 o'clock. I thought it
No,
is.
sir.
No, 8
Is there
o'clock.
any specific difference between cold pasteurization and simple pasteurization? And cold pasteurization is limited to liquid foods only.
No, it can be used for the solid food also, like a cold plasma is there.
Okay.
Right? Radiation
And
ionization is
someone
there.
has asked that best technique for storage of millets.
Yeah, storage of millet actually is the question mark with reference to the rancidity actually.
Yes, sir.
So once it is actually processed, then actually is not the issue. But if you store, the rancidity of the oil content is actually the question mark,
Okay.
for the millet. Yes.
Okay. And then one question from Dr. Shivani Sharma, ma'am. If a small residue of hydrogen peroxide remains in milk,
Yes.
it can be harmful or alter milk compositions.
Yeah, it can be harmful actually, but then because when we are using, why we are using the catalyst to remove that residual hydrogen peroxide? That is the purpose. Actually, our work was on the catalyst. But what is the application of that catalyst? To remove the hydrogen peroxide. Why? Where we are using hydrogen peroxide, we are using for the cold pasteurization. That was the answer, yes.
If any questions from the participants, you can unmute yourself and ask, sir.
Definitely.
One or two questions can be taken.
Yeah, I didn't realize. I thought my time is still 8.
Sir, everyone was listening to you, and it was a very nice talk from your side. If any questions, please, you can unmute yourself and ask a question.
Yeah, definitely, because I am here only for some next few minutes only. Then you people have to get ready for the next
Good evening, sir.
Yes, sir.
Yeah.
So what about the technique cryopreservation, like when we just store it in liquid nitrogen at a minus 196 degree?
Yeah, exactly.
Like, is it used in the industry for preserving the samples for the IE life shelf?
No, actually, it will be very expensive, no? Most of this cold pasteurization process are that engineering aspect which I was telling, no? Are very expensive actually. Like, for the cell line, bacterial cultures, organs, you can
but we do teach in cryopreservation, like they are preserving the pollen grains and preserving some samples.
The seed material, the bacterial cultures, the cell line, we can actually store in that one. But then we can't afford to store the food grains, no? Because food grains, in 100 rupees you will get 2-3 kg. You store that one in a refrigerator, how much will be the cost of that, no? So it will be work out to be expensive, and that's why this some of these methods which I was telling, cold plasma, found to be very actually expensive, or high pressure preservations, found to be expensive actually, works out to be expensive. But when it is done in a bulk, in a large scale, in industrial scale, it is definitely economical. And now in Europe, US, they are using. I don't think the milk which I was getting was boiled like how we boiled in our Indian industries, dairy industry. Yes, I have not paid that attention somehow. But then we get gallons of milk there. I don't think they use the method of what in India we are using, the pasteurization method. That is what I wanted to tell.
Okay, sir. Got a clarity, sir. Yeah. Thank you.
Thank you. Thank you.
Thank you so much, sir, for your expertise and valuable guidance which you have provided to our participants with a deeper understanding of modern preservation methods and their industrial applications. The clarity with which you addressed their queries and the practical perspectives you shared will serve as a lasting inspiration for researchers, academicians, and entrepreneurs alike. The emerging role of bacteriocins, the ribosomally synthesized antimicrobial peptide produced by lactic acid bacteria and other microbes as a safe and natural biopreservative in the food systems, were emphasized very nicely by you, sir. You explained bacteriocins such as lysine, pyriocin, and enterocin inhibit spoilage microorganisms and foodborne pathogens without affecting the sensory quality of the food products. And the synergy with modern technologies like cold pasteurization and hurdle technology further enhances the effectiveness in post-harvest good food management. We deeply appreciate the intellectual depth, empirical evidence, and practical perspectives you shared, which have enriched this FDP and created new avenues for collaborative research, academic inquiry, and technological advancement. Your engagement with participants during the discussion and question and answering session further enhance the scholarly value of this session. We are truly honored to have you with us, and we look forward to your continued association and guidance in the future with us. On behalf of Swami Shri Swarupanand Saraswati Mahavidyalaya Bhilai and the organizing committee of ATAL Faculty Development Program, I, the coordinator, extend my profound gratitude to Professor Prakash Halami Sir, Chief Scientist, Mysuru, for his scholarly lecture on post-harvest technologies, biopreservation, and cold pasteurization. Thank you so much, sir.
Thank you so much, sir.
Thank you, everyone. Yeah, thank you.
Yes, sir. Yes,
Bye.
sir.
Thank you.
Thank
Now let's start our fourth session.
Has sir joined?
As a join, I didn't see.
Dabai sir
the-
has he joined?
No.
Otherwise, I have to ask him. Let me
Please,
ask.
ma'am. Please, ma'am. Ask.
Yes.
Sir, ma'am, sir is there, no? Sir is
Sir
there.
is there? Okay. So, good afternoon, Dabai sir, because it's afternoon there. We are
Okay.
here where it is evening, but you are there with a good afternoon. So, a very good afternoon to you, sir. And for you, I should start that science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. The sentence given by the Louis Pasteur. With this thought, it is my privilege to introduce Dr. Aliyu Ibrahim Dabai, a distinguished scientist whose work exemplifies this spirit of science without boundaries. We are honored to introduce Dr. Aliyu Ibrahim Dabai, currently serving as Higher Scientific Officer at the Agri-Food and Biosciences Institute, Belfast, UK. Dr. Dabai holds a PhD in molecular microbiology and metagenomics from Queen's University, Belfast, UK, with extensive expertise in applied microbiology, recombinant DNA technology, biocatalysis, and bioinformatics. His professional journey spans over 17 years in academia and industry, with specialization in soil microbiology, metagenomics, microbial biotechnology, and bioprocess development. He has worked as a postdoctoral research assistant at Queen's University Belfast and LMAC Sciences UK, where he developed customized recombinant protein expression system for industrial applications. Previously, he was a senior lecturer at Usmanu Ranfodio University, Sokata, Nigeria, mentoring undergraduates and postgraduate students and contributing to curriculum development. Dr. Dabai has successfully supervised 20 postgraduate students and over 100 undergraduates. His research contributions are supported by prestigious grants, including the Global Challenge Research Fund, UKRI, OPCW Plant Biomarker Challenge, and TETF Fund, Nigeria, focusing on soil health, bioremediation, and sustainable agriculture. He has published widely in high-impact journals, contributed book chapters, and participated in international conferences such as World Microbial Forum, African Agriculture Science Week, and Molecular Microbiology Ecology MMEG meetings. Dr. Dabai is also a member of leading professional bodies, including the Royal Society of Biology, UK, American Society for Microbiology, Applied Microbiology, International and Nigerian Society for Microbiology. We warmly welcome Dr. Dabai and look forward to his valuable insights on topic sustainable farming practices at this FDP. Last but not least, I would like to thank Dr. Deshmukh for introducing Aliyu Dabai for this FDP. Sir, the mic is all yours. Please proceed.
Thank you very much, Dr. Sharma, for the elaborate introduction and for your effort to get me on the conference, because if not for you, I wouldn't be here.
No, it was
yes,
actually
I'm grateful.
Sir who introduced me to you.
Yes. So I'm very glad to be here, and very good afternoon from this part of the world to everybody who is attending the conference. And I can see it's widely, attended. I can see the number of people. It's encouraging, and I'm glad to be part of this. As Dr. Sharma has introduced, I will, be talking about sustainable farming practices and bringing in a lot of experience that I've generated across Africa and Europe with respect to sustainability and farming practice and, the agric environment. As we dive into the slides, feel free at any point if you have questions, you can ask, you can drop it, and I will be glad to respond to all of them at the end of the session, as the organizers may deem necessary. Thank you very much, and a very good afternoon all. So I passed the slide, Dr. Sharma. I don't know if you have it.
Yes. We are having your slide. So we have to open it from our end, sir. We have to present it from our end.
Okay, let me see if I can open it from here.
Rupali ma'am, I have mailed you. If you want, I can send you in the WhatsApp also.
Okay.
Yes, ma'am, I'm checking.
Otherwise, I will be sending you in the WhatsApp also.
I'm checking this. Just wait.
Okay, I think I'll get it.
Shared in WhatsApp also, ma'am, to you.
Okay.
Can you see my screen?
Yes, sir.
Okay. Yeah, I think that's us then. I can proceed if everyone is happy enough. Yes, can I proceed?
Yes, sir.
Yes.
Okay. So I would be giving my talk today on sustainable farming practices, which I have subtitled it as Cultivating a Greener Future, which is part of the AICTE and ATAL FDA conference on technological innovations and agri-tech and food processing. As Dr. Sharma said, my name is Ibrahim Aliyu Dabai. She's given a detailed introduction there, but I work for the Agric Environment Branch in Agric Food and Bioscience Institute in Belfast, in the United Kingdom. Belfast is part of Northern Ireland, which is one of the countries that form the United Kingdom. And I also hold a visiting researcher position at Queen's University, which is where I actually did my PhD. So,
can we make it full?
hello?
Yes, sir.
The slide is not moving. Okay.
Is it now?
It's not.
Okay, let us present that. You please stop presenting it. We will be presenting.
Okay.
Rupali ma'am, please. Request to all the participants to please mute yourself.
Yes. Okay. We'll just take it from there. So if you move to the next slide. So we've seen the topic, we're talking about sustainable farming practice. But when you talk about sustainable farming practice, we have to know what is sustainability in the first instance. So if we go by the definition of the Greenpeace, which is a non-governmental organization here in the UK, you know, we'll be asking a couple of questions in the first instance. We know that most environmental problems, they come from natural sources, which we use unsustainably, right? And when we then look at sustainability, we would now say it's a way of using resources that will allow us to continue using that forever. One typical example would be renewable energy. You have the solar energy, you have the wind energy. So all of these are sustainable. They are renewable. You can continue using them. And of major importance is they do not have an adverse environmental concern. So when we talk about sustainability, we can also split that into two. You can say sustain and enable. When we say sustain or sustain/able, or we say an activity is sustain, or you want to sustain an activity, basically you're saying that that activity is able to be sustained, if we know the definition of sustained, and without running out of resources or causing harm to either the environment or human, or any other creation, living creation or the ecosystem. So, but when we look at the act of using up resources faster than we had thought, there was all of this hypothesis: you have resources, and this resource is going to, do all of the entirety of whatever time that humanity has got on Earth. What we are now seeing, because of technological development, all of these resources are depleting faster than they have been hypothesized. And if that, at the pace we're going, that is an unsustainable practice. It means the world is going to come to a stage whereby we'll be reaping the adverse effect of some of our actions. So sustainability, if you look at it in a few other contexts, they may have different meaning depending on where you're looking at them. I also do a bit of ISO standard, or I'm a ISO certification lead auditor. So again, if an auditor is looking at that, or somebody come from ISO standard, when they talk about sustainability, they may mean something. But in the context of this talk of sustainable farming practice, most of the things I'll be talking about here is referring to environmental sustainability. But what about financial sustainability? What about social sustainability? All of these ones, also got their own definition. But like I said, in this talk, we would, majorly look at environmental sustainability, because that is the context in which our farming practices fall. Next slide. So back slide. A slide has been skipped. Yep. So when we talk about why is sustainability important, why do we need sustainable farming practice? So when we talk about sustainability and why we think this is important, it comes to the concept of we, as human, trying to fight climate change, trying to protect our environment so that we can live a healthy and sustainable life, right? Because we're all seeing this problem. Years back, if you take 20, 50 years ago, the things we're seeing now, we're not seeing them, ranging from flood, to extreme heat, India, like Nigeria, where I come from, we're not exemption. In the developed world, some of these problems, are even, more devastating, as we've seen in recent years. So the things that come to mind when you talk about climate change, you're looking at environmental problems, and what are these problems, or what's contributing to these problems? You have deforestation. We're cutting wood unsustainably to make timber, to make all sort of furnitures at home. We are losing biodiversity. We are, there's problem of overfishing. Then you got a big problem, which is pollution. Because of industrialization, now you have different kind of pollution. You have the plastic, you have pesticides, which is coming from agric, which is relevant to this talk. And then you got hydrocarbons, because we got, farm machines and the like. And then you have heavy metals. Each of these ones, we know what, effect they pose to us as humans. So what is important is our ability to come up with actions that are natural, that can mitigate the impact of these, adverse environmental problems. If we can't do away with them, then we have to come up with ways to try and reduce that. And, a solution to doing that is that we have to do things in a sustainable way. I mean, we've seen that there are things that could be avoided, but, based on human actions, some countries feel they are more developed than others. They are using more natural resources, or are contributing to more to climate change than other countries. And now we're seeing things like carbon footprint, carbon accounting. What does this mean for developed countries, and what does it mean for developing countries? Right? Nigeria produces oil. India is one of the huge, or a big friend of Nigeria that imports oil. So if we put all of this together and put that in the context, who are the big users of these natural resources? Who are the suppliers? So when you come to do your carbon accounting, or monitoring carbon footprint, the product that is produced in one region may end up completing the life cycle in another region. So all of those things come to play. Next slide. So when we look at the pillars of sustainability now, what are the pillars? There are key things that are important to look at, but basically, when you talk about the pillars of sustainability, the 3 P's come to play, because these 3 factors are driving sustainability. One, you have the planet, which is the environment where we find ourselves, where we interact. 2, you have the people who want to live large, right? Who want to explore all natural resources. And then you have profit. Companies want to make more money, right? But in the middle of all of that, you've got the sustainability, which is striking a balance between the 3 P's. And these 3 P's are the core foundation of the framework of the United Nations Sustainable Development Goals. Now, if we know that, we know it's a big deal. How do we leave a sustainable planet? What are we leaving? What legacy are we leaving behind? Yes. Next slide. So, what do we need a sustainable farming practice? Haven't looked at what sustainability is and how we have to be sustainably conscious. We have to also know, why do we need a sustainable farming, practice? Next slide. So, we know the world population is growing. As of today, the global population surpasses 8 billion. And then there's huge demand for food, which is placing an unprecedented pressure on agriculture. Farmers want to farm, they want to make more money. Yet, we know that the conventional farming methods can contribute only X amount. And because we've been doing that over and over and over again, the same kind of practice over from, generation to generation, we're passing that. What we are now seeing in recent times is that conventional farming approaches have resulted to our soils being degraded. They've resulted to water pollution. You have greenhouse gas emissions. So if you put all of this problem, you don't need to be a scientist to see them. You don't need to be a scientist to feel the impact. One thing is clear, that we then need an urgent solution that is resilient and environmentally responsible way to try and, produce food to meet the growing world population, and reduce or slow this situation. A lot of the rice that is produced in India end up in Nigeria, because for Nigerians, the sign of your class, right? Your class or your status depends on how much rice is eaten at your house. So most of the rice produced in India then end up in Nigeria. So you see, that's one thing when I talk about the carbon footprint. So when we look at the two pictures here, on this side you have, on the left side you have the degraded land, and then on the right side you have a restore land, and we can see the difference in terms of the rice that is planted on the field. Next slide. So when we talk about that, we'll talk about soil degradation now. So that brings us to a very important concept, and that is the whole, everything I do here in the UK, I was basically employed to set up that lab, which is known as the Soil Health Lab. So it's a lab that I'm still setting it up, but we've seen a lot of progress. So we would have to look at what is soil health. And then here, it focuses on maintaining the soil structure, soil fertility, and then microbial diversity that will support a healthy and a resilient crop production. This is, when we talk about microbial ecology, there's a lot that takes place, and whatever happened below ground biomass has an impact on above ground. What will happen to a crop above, above the ground is usually the solution to it is under the ground. So if you can correct that, or if you can maintain that healthy soil, then you're on your way to making a productive and a sustainable production of whatever crops that you are harvesting in that or you intend to harvest in that particular land. Next slide. Then the next thing is sustainable water management. Water is becoming an issue. Where I come from in Nigeria, which is in the northern part of Nigeria, years back, we look at Niger and Libya, the Sahara Desert. We call them desert, but that is encroaching now, and that is biting into Nigeria. Lands that were previously cultivated, we can no longer cultivate it now. The soils are becoming drier. So we need to come up with innovative way that are efficient, so we can irrigate our land, we can use that, and then that has impact also on reducing the pollution of this water, because sometimes you may find the water but it's polluted, and then when you use that, that has impact on the yield, or the end product, which is the produce that you are going to harvest from the farm. Next slide. Then the next thing is biodiversity. Biodiversity is there for a reason, and they have been keeping our planet together. And once you disrupt the biodiversity, you're equally disrupting the ecosystem. And the consequences is what we are seeing now as climate change, or as the outcome of climate change. And when we talk about promoting biodiversity, we're saying you encourage natural ways of doing things. Talking about pest control, talking about pollination, because some of these agrochemicals that we use today have been shown to reduce bee populations, to reduce the pollination, and we know what they do in pollinating within the fields. Next slide. What about integrated pest management? A lot of use of synthetic pesticides, which is a big problem that I found, especially in developing country. These companies are pushing this thing to them, but nobody's looking at the adverse effect. They're just putting a lot of chemicals. Take it there. This is herbicide. This is a different kind of them, but they all have effect on the microbial biodiversity. And once you disrupt that ecosystem, you are creating a potential problem. It might take time, but it's certainly going to happen. So we have to look at ways that we can reduce the use of pesticides. That will mean either by crop rotation, and then I'll look at that once we come into each slide, and using typical examples of some of the things that I am managing here in the UK. Maybe some, especially with us, developing country, we have some cultural practices and that, are great-grandfathers have been using, and it's been working. That is before the mechanized agriculture came in, and then they told us you could use chemical, you could use synthetic, or hybrid seeds. At the moment, I'll give you an example. South Africa is in a big problem that they don't have natural seeds, because they've solely depend on, the hybrid seeds that now, that is creating dependence, which means every year you have to go to this company to buy seeds to plant. But where we're coming from, in the past, farmers would plant seeds and then usually keep effluent, and that has been passed from generation to generation. And you never have, reduced yield. But suddenly we have the impact of climate change. Some other factors are responsible for the reduced yield. And then, farmers just started jumping to the hybrid. Yes, hybrid will help solve the problem. But if we're talking about a sustainable way of doing that, we might have to have a rethink if that's the way, we are going at the moment. Next slide. Reduce input as well. Amount of fertilizer you're putting in, that has been linked to runoff. They go and pollute the nearby water bodies. That makes the cost of treating water more expensive. In Northern Ireland, where I am here, there's a big problem. We have a river that's called Lough Neagh. The Lough Neagh is an important river in Northern Ireland, and that has been an issue. There's smell coming from there. There's cyanobacteria and algae have covered the whole of the water. That it is creating serious concerns within government cycles. And I can say within the organization I work, we have more than 5 different kind of grants this year alone in trying to find solution to clean up that problem. But this is not a problem that just happened overnight. It is a series of problems that have compounded over the years. Based on political reasons, nobody want to talk about that. But, out of this talk, let's leave the politics out of that place. Next slide. So, soil health. This is what I do. This is like my bread and butter. I'm living a life in the UK based on soil health. And when we talk about soil health, we're talking about an healthy soil that is important for the sustainability and productivity of agricultural systems. That is what we would term as, a healthy soil. And when we talk about a healthy soil, it's simply the ability of a soil to continuously have the ability to function as a vital ecosystem for sustaining plant, animals, and humans. And we're seeing that in recent time, this is not the case. And in doing that, there's a whole lot of factors that come to play. You're looking at integrated livestock, you're looking at eliminating chemical disturbance or tilling or no tilling. You're looking about appropriate, biological disturbance so you don't disturb the ecosystem. You're looking about, providing and maintaining cover crops. We'll see that in details, and then maximizing plant diversity or microbial diversity, and then you continuously keep that cycle so that there are roots in there, you have earthworms, all of that, performing their role. They're there for a reason. And if they're not important, nature is not going to have them there. So in UK, there are, some sustainable farming practices that are in place. Some of these, we're looking at the cover croppings, where you use either a deep-rooted plant versus a shallow root, and then you rotate the crop. The idea is when you have a very deep-rooted crop, it means they can access the nutrients that are deep down. And then you have a shallow one, they died, and then the biomass decompose, then they add in the carbon. So that brings up all of the issue of, carbon sequestration in trying to improve climate change. And then when you do that, cover cropping, it will help you put the carbon down. In a way, you're promoting carbon sequestration. Then there are instances when they will help as well to promote weed. Some of them have, they spread easily. They would then shadow weeds that are not wanted in the farm. So those weeds would then get, because they cannot access nutrient, due to competition, they die, and then their biomass is fed back into the soil, and then that will increase soil organic matter. Again, some of them will all help to suppress disease, because some of the plant disease that we have, these cover crops aren't susceptible to them. So it meant when you have a copper cover crop, the pathogen cannot have a susceptible host, because we know as microbiology, you need a host. A disease cannot propagate if there's if you don't have a suitable host. So when you do this cover cropping, it's been shown that, again, that will help you suppress disease, and then you would fight the issue. You would improve the soil structure, and then, there won't be any time of the year where the soil would be empty or barren, and then promoting erosion. So technically, that will help you solve the issue of improve soil structure and then reduce erosion. Then you have no tillage. In different part of the world, the till. Where I come from, we till. We do a lot of tillage. Mechanized agriculture is the same thing. You use the soil. Once you're disrupting the ecosystem, you're tilling the soil. You're breaking roots. And these roots are important, to keep the soil structure intact, so that they can, they can promote whatever biodiversity is there. So if you want to improve, soil, you probably have to go on, and then there's a lot of experiment here in the UK that shows that this is actually working, for arable land. In Northern Ireland, it's a bit different, because 95% of the land in Northern Ireland, they are not arable crops. Yes, they grow Irish, what we call the potato, not the sweet potato, whatever one we call it, Irish potato in Nigeria. But 95% or close to 95% is used for cultivating grasslands, and these grasslands are then fed to animals. And that's why I think the major agricultural sector that is of importance here is the beef, production industry here. They're very strong, and then they're shipping. A lot of that goes into Europe market. And then you have organic soil enrichment, where you use maybe farmyard manure or animal compost, or vegetables, and to sort of boost soil fertility and structure. And all of this then fits into the idea of trying to improve carbon sequestration, or you boost the health or a healthy soil. Next slide. So this is just a typical example. If I give you a landscape here, this is just the nitrogen cycle. As microbiologists, we understand the role of the nitrogen cycle. I'm not going to say a lot about this, but these are the individual components where you have, atmospheric nitrogen, or you have fertilizer added to the soil. The animals, they pass their liters, and then that goes in. You have volatilizations to things like ammonium, and all of that is emitted into the environment. Nitrous oxide is a problem, as part of the gases that we're monitoring here where I work, looking at greenhouse gases. So we look at CO2, we look at nitrous oxide and methane. So all of these are the gases that we're producing. This complex structure here shows how all of these are linked together. They're all interrelated. So if you disrupt one of them, then you are creating a loop within this whole network. So the way nature has kept this network is important for the sustainability of the whole system or the whole farming settings. Next slide. So how do we monitor that? We'll talk about healthy soil, healthy soil, healthy soil. Yes, we need a healthy soil to maintain productivity and sustainability within the farming system. But how do we maintain that? How do I know that my farm or my soil or my farming setting, the soil in my farm is a healthy soil, or my farm is healthy, and as such, they can support the growth of plant and other living things that are found within that setting? So when we're monitoring soil health, there are 3 components that are important, and each of these ones play a role in the health of the soil. So we have components which are physical, and then we have the chemical component of the farm, and then we have the biological component. And in the middle of that, that's where we have the soil. When you have a balance between that. If you remember when we talked about the 3 P's of sustainability, at the middle of that, that's where we have a healthy soil. So but when we talk about the physical now, what are we talking about? We're looking at soil structure. We're looking at bulk density. We're looking at microporosity. We're looking at mesoporosity. We're looking at microporosity, water capacity, residual porosity. We're looking at potential resistance, saturated conductivity or dry aggregate, wet aggregate, surface hardness, and the likes. And when we look about the biological, so this is basically what I do. We're looking about, root pathogen pressure assessment. How is it likely that your root are going to be assessed? Beneficial nematode. And there's something I've developed along this line, which will tell us, because we have different kind of nematode. You have those that feed on bacteria and those that feed on fungi. So depending on the ratio of the two, you can actually predict the health of your soil, which is one of the things that I'm developing now. It's still in its early stage, but what you tend to find, if you find more fungi feeding nematodes versus that of bacteria, that has implication for carbon sequestration of in that particular soil. Then we have potentially mineralizable carbon and nitrogen. We have cellulose degradation rate, and then we have the particulate matter, we have active carbon, and then we have the weed seed bank, we have microbial respiration. Microbial respiration is still the same thing as potentially mineralizable carbon. We'll see that in my next slide. And then we have soil protein index, then we have the organic matter. On the chemical side, these are the commonly used ones. We look for soil phosphorus, soil nitrogen, soil potassium, pH, magnesium, calcium, iron, aluminium, and the like. And each of these ones depend on the level in the soil. You would have to do that. But having said all of this, there are a couple of questions that we need to ask. Number one, which one are you going to look for if you were to assess your soil? What are you going to look for? Are you going to do all of them? All of the biological, all of the physical, all of the chemical? Because this is going to cost farmers, and this is one of the reasons why farmers are not adopting it. And in the last ISME, which is the International Society for Microbial Ecology, this was some of the things. We're talking about climate change. Microorganisms are a key player in all of the, the cycle. You talk about carbon cycle, nitrogen cycle, phosphorus cycle. Microorganisms are the key players. But microbiologists were not being invited. Microorganisms were not being, added.
Only microbiological health can tell about the soil health. Alone, microbiological test can tell us about the soil health. No need of the
so that's where
test.
Yes, that's where we're going. I would say that's what I do. So the next question is, if you don't know, if you don't have a baseline data, how do you tell that your soil is healthy now? Say, for example, I ask you to pick a soil. You need a background data. You need a baseline. Years back or 2 years ago, this is what we used to have. And then after 2 years, something has changed. That will be an initial trigger to say you need to do something about your soil. But most times we don't have that data. So this is where the problem is. You don't have a baseline data, so you don't know. So when they say soil health, you are thinking in your head, sure, I cannot do all of these things, or it's going to cost me money, especially for commercial farmers. It's going to cost money. So you can't keep doing all of this each year. Next slide. So the Soil Health Institute, which is based in the US, there's another one here in the UK, it's called ADH, something like that. And they are doing this. They're trying to build out, a baseline for each country. But unfortunately, what I find is most developing countries are not there yet. They're not building this database. You can't use information from the US to sort of predict your own soil health, to know if your farming, sustainable farming practices are working. You need this baseline. So most developing countries aren't doing this yet, but hopefully they will be there in the next couple of years. So what the Soil Health Institute have done is they have sampled 100 scientists across 124 locations or research stations in the US, Canada, and Mexico. And they have evaluated about 30 of these, soil health indicators that I've mentioned in the earlier slide to try and come up with what is the minimum information that I need that could tell me, something about my soil health. And, these are some of the outcome that they have processed. And at the end of that study, which was just released not long ago, the recommendation is the things that scientists found very useful in their research farms is the soil organic carbon, potentially mineralized carbon or microbial respiration, and then you have the aggregate stability. Now, this aggregate stability, it's very easy to do. And then now there's a mobile app. The app is called Slates. I have it on my mobile. It's just an easy way to tell you the soil aggregate, which has a value between 0 and 1, depending on that. So if you see a reduction in that, that is an early trigger to say, something needs to be done. And then you can then begin to look at the other variables. Other than that, these 3 should be enough to look at that. Having said that, this depends on location. This study was done in the US. So I cannot close my eye and say the same thing is applicable to India, or the same thing is applicable to Nigeria, because there are practices that we do on our soil that might impact the overall biodiversity of our soil. They might be different because of certain practices that we do, which may be cultural or may be part of our farming practices. Next slide. Next slide. A slide has been skipped. Yes. So part of the things I do here, which is still in the early stage, is I'm trying to see now as a microbiologist, can we develop one approach, one or a couple of them, that we can use to have an idea of what is happening in the soil. And the whole idea of this is to position microbiologists as key player in the field of sustainability. Because microbiologists are important. Microorganisms are doing all of these things, but we're not being put in the loop. Next slide. The slide before this, not next, this previous slide. Let's just jump to. Yes. So as part of the things I do here in the soil health lab in the agric environment, what we're doing is we're using microbiome science or soil microbiome, through metagenomics and qPCR. And what I'm hoping to do here is I want to come up with an approach, because the agency I work for, they are like the research component of the Department of Agriculture, which is like the Ministry of Agriculture and Rural Development. So we interact with farmers, we conduct research, and then we provide policy direction for the government. So that is what the government then use. Because government is talking about now, at the moment, they pay farmers for their carbon, what's that? Carbon, they do carbon accounting, and then they pay farmers. And then these farmers get subsidy. So what the government is trying to do now is to tie that to their carbon footprint. The lower your carbon footprint, the more money you get. So that, but that is making farmers cut their old ways of doing things. So, but government will need an instrument to be able to measure that, so that they don't rely on farmers. So from time to time, they can go and take soil and measure that and say, yes, we know what is happening based on this. So my job is to develop that. And I am kind of basing that at the moment around functional nutrient cycling, which is C, N, and P. And that how that can then help us for sustainable nutrient management. And the approach as well, I'm making it in such a way that it is scalable, it is cost effective, and it has a quick turnaround. Because now what they have at the moment now is, even if they bring the sample in-house to do all of the soil chemistry, it takes a lot of time to do that. And when you're dealing with a lot of samples, you have every farmer trying to do that, because the money they pay, the government pays them, is done at certain time of the year. So all of your carbon footprint have to go to be submitted to the government. So assuming every farmer is rushing to get this information, what happened then? You have the carryover. So the government is looking for something is quick. We can do it quickly and then have an answer to that, and then that can be deployed to other national and international farming systems to try and provide solution. So on the lower side, where we are, or one of the take-home message at the moment, I have developed something that has shown strong correlation between soil P, S, and soil protein index. So you've seen all of the different variables up there. So now, from one of the things I developed, one of them can tell us information about the P, the S, and soil protein. And then the second one can tell us information about total nitrogen, active carbon, PMC, and that had, all soil organic carbon, and then soil pH, soil K, and it had a negative relationship with magnesium. So this is just one gene out of the several thousand of microbial genes that I could be looking for. But this gene has given us this information. What is the take-home message here? The take-home message here is you could target this gene of microorganism to sort of provide an early direction for a farmer or for a policy maker to sort of gauge any of these variables here without the need to do all of the chemistry in trying to provide answers for each of these ones, because each of these ones will take days before the soil health lab can give you information about each of these. But with this one gene, you can collect soil today, isolate metagenomic DNA today, and get result either today or tomorrow, depending on the amount of sample. But what I found out is within my capacity, I can process 20 to 30 samples within 2 to 3 days, and the result is out. And so far, with the exception of pH, most of those ones require the samples to be processed before you could even think of any of those variables. Whereas with what I have developed, you don't need to process the sample, but the samples can be stored for use in -80 or -20 for short-time use, but you don't need any processing. Get the sample, isolate your DNA, and you're ready to go. Whereas every other thing there, you would need to do something in terms of processing the sample, either sieving or something, any of that, that may take time before you have a result. Next slide. So this is one of the ones that I have done. So I'll just be brief. What is available in the literature is when you're looking at microbial respiration, or it's called potentially mineralizable carbon, which is the amount of carbon, because within the soil organic matter or soil organic carbon, not all of the carbon there is available for microbial metabolism. When we look at the active carbon, these are the carbon that are easily metabolizable for microorganisms to be used to promote their biomass. And then when we talk about PMC, we're looking about microbial respiration now. And the idea is the population can be directly proportional to the microbial diversity within that system. So usually there are different ways you could do that. Basically, you incubate a fixed amount, which is usually 3 grams of sieved and air dry soil, and usually you incubate that. So the idea is when you incubate, most studies would put what is known as CO2 trap, and this is basically calcium carbonate, KCO2. And the idea is what you're trying to do is if the CO2 is produced from microbial respiration over time, so you could do that 24 hours or 48 hours, 72 hours, or 1 week. When they produce that carbon, it goes into that trap, which is an alkaline solution. And in the end, you can now use titration or whatever chemical approach to look at that. But one thing I found out when I started is that with microbial activity, it's not like it's got a switch. The fascinating about microorganisms, everything they're doing, they somehow don't know they're doing that. All they know is they've got to devise a way to survive. So it's a case of survival. They have to survive. If you make the environment harsh, they're going to survive anyway. So once you start the experiment, you cannot stop it. So what I find is the trap gets saturated. So if I come after 24 hours and take the amount of carbon there and measure it, I may be underestimating it, because that solution can, depending on the molarity of my solution. So usually they use something like 0.25 molar of KCO3. So if you use that, K2CO3, so if you use that and it's saturated, say, after 12 hours, and then I come after 24 hours to monitor my carbon, I wouldn't see that excess. So that will now result to underestimation. So what I have done here, I designed a system where the soil is incubated inside, and then that is sealed. And then I have a valve here, which I still have there. I'm watching it here in my office. So you incubate that. With this, you don't have to worry. Just incubate it, and you can use a syringe and needle to take out your gas from this. And once that gas is taken, it's taken to this bottle. And these bottles have been pre-evacuated so that there's no air. There's, all of the gas has been, usually we pump nitrogen here so that every other lighter gas is taken out. And once we have this injected, so you take like 20 mil, that tube here goes in here. But this tube is just about 12 mil. So which means when you inject the 20 mil, you're going to have some left. But you're better off taking 20 mil. And then that is then ejected into a GCMS system. Again, everything about GSM, we know is sensitive, but then you need a standard. So we have the CO2 standard, the C4, and then the nitric oxide. So all of these three gases can be measured. From there, we can use that to calculate, putting it in this formula, we can work out how much of CO2 is there. And we can then convert that to gram of soil. And simply what you do is, whatever value you get from the headspace out of this formula, you can multiply that by the volume of this chamber which you use in your incubation, and then divide it by the amount of soil. So in my case, I use 3 grams. So that, and the incubation period, if you do it for 24 hours, then you just divide by 24. If you do it for longer, then you use that to divide. And what you get at the end is going to be a milligram per CO2 of carbon per kg of soil. And then that is a value that is then used as indicator of the soil health. So if you already know the background, then this will provide you information to know whether your soil has improved in microbial respiration or it has reduced. But if you don't have the background, then that is where it becomes a bit tricky. Then the question is, what do I do with that information? So that's why the qPCR approach that I have developed based on metagenomics or soil microbiome is there to help with this. And we're seeing in the earlier slide that there was a relationship between certain microbial gene and the microbial recession, or what is known as PMC, potential minimizable carbon. Next slide. So then that brings us to, sustainable water management strategy. So this is one way that we could ensure, a sustainable farming practice. When we talk about water management, you have to look at efficient way, and basically, you may have to resort to precision methods like drip irrigation or micro sprinklers, which you put, in fruit farm. Basically, the whole idea is you are trying to reduce water wastage and then improve the quality of your crops. Rainwater harvesting, this I think is something that I've read is very common in India as well. In Nigeria, they don't do that a lot. But one thing is you could harvest the rainwater. Here in the UK as well, it's not something that's easily practiced. But rainwater is an option where you just put insulation to harvest the rainwater in your building, and then that can be collected and stored in underground reservoirs. Basically what you're doing here, you are reducing reliance on the main water supply. In Northern Ireland, unlike the other part of UK, people pay water bills, but in Northern Ireland we don't pay. But if you are using the main water supply in your farm, the government will arrest you. And in a couple of years, not long ago, somebody has been arrested and they have been jailed. If you use it in your farm, then you're supposed to pay for water bill. Other than that, Northern Ireland, there's no water bill. But the rest of the UK, yes, you have to pay. If you leave, you have to pay the water bill. That's one way, they could harvest the water and use it. But because we're surrounded, it's an island, we're surrounded by water. So nobody's really bothered about harvesting rainwater. And then you have efficient, water efficient crops that only need little amount of water. I know this has been done with sorghum, which is one of the common food that we use in Nigeria. And I think India as well have that, which is of different color. Wheat as well that require, and then barley, and we have them here. We have. So in my experimental farm last year, coming to this year, I had oats in the farm, and then I had winter barley, and this is barley that tolerate cold when we have snow around. So those barleys are meant to survive. So they were only harvested in June, at the first week of June. That's why I've harvested those ones. So that's one option to go. And then you have the integrated approach, which is you're combining irrigation efficiency with rainwater use and crop selection. The whole idea about this is the long-term sustainability of water in your farming system. Next slide. So these are just examples of the various water management systems that have been implemented globally. You could see that, you have the sprinklers, and then you have all of that there. So you could see different approaches where you could use to supply water on your farm. All of this is aimed towards sustainable use of water and promoting crop quality and yield. Next slide. We'll talk about biodiversity and integrated pest management now. Like I've shown you with the nitrogen cycle, you can see there's a lot at play in ensuring an efficient nitrogen cycle. So whatever you do in your farm has potential to disrupt the nitrogen cycle. And we know nitrogen is important for plant. We know P, which is phosphorus, is important as well. We know carbon is important. So what you do in the farm, is very important in trying to, promote the biodiversity in the farm. And so in the UK, we have things which we call hedgerows, and these are usually at the edge of the farms, or what we call buffer strips. And these basically, they provide like some sort of natural habitat, which are beneficial to keep insects within your farm, so they can help with pollination. They also keep wildlife. There's a lot of that in the UK that you see, especially, some of them are supported by farmers. And then, there are a lot of NGOs which are supporting farmers, to sort of put this in place to promote, biodiversity protection. Then you have the crop protection, which I have mentioned earlier. And the idea about this, you want to disrupt pest and disease cycles. But then you put one crop that is not susceptible to the pest. You take care of the pest because they don't have where to attach, and then cause disease. And when they can't attach, they can't multiply, they can cause infection. And by the time you bring their host, it would have been the other part of the day. So the pathogen population would have gone, very low. And the whole idea about this is that when you have an healthy soil, the healthy soil is supposed to be able to protect itself against, disease-causing plant pathogens. And then there are a lot of them here in the UK and in China as well, which are put in place to protect nematodes. Just from where I'm seated here, there's a branch that we have within this organization. I don't work there, but that's just their job. Their job is to set up this crop protection, because if you remember the Irish famine that they had years back, that a lot of them moved to US, that's what happened, because that nematode destroyed all of the potato, and then there was a big hunger crisis here that wiped a lot of Irish population, if you remember that. So that led to a lot of them moving to Canada, America, and different part of the world. So that's why they have to keep abreast in trying to stop that from happening. Then we have biological pest control. This is where we use natural predators, like microorganisms, parasites, wasps, and predatory termites, to be able to control pathogens within the farm without the need of using agrochemicals. But unfortunately, the agrochemical industry, they are also not sleeping. They are also trying to promote their business without minding the global consequences of these chemicals. Then we have the global organic pesticide. These ones are the microbia, which have been, they are different level in developing countries. It's still not there, but Bacillus thuringiensis is one thing that you can use. And this, once ingested by the host, it sort of disrupt the gut, and then that's the insect is gone. And then, instead of using pesticides, and then you have the integrated approach, which basically you maintain your habitat, try and make sure that your farm is as natural as possible so that it can support itself. Because when you have a natural system, they have that resilient ability to withstand all of these different changes, either from physical, biological, or chemical means. Next slide. Yeah, so that's one typical example, where you have a pest, and then things you do to reduce that. So here you have the, crop rotation. You put a legume versus that. So a pesticide or a pathogen that affects that may not affect that. So by the time you rotate between the two crops or more crops, that will help you take care of that. And this is practiced, in different part of the UK. So this is one typical pictures that you find here in the UK. This is from one of our experimental farms. So this is what you see. Then you have all of the strips. These strips here can help to trap when nutrient is flow, because you see this up here is hilly. Usually there'll be, water flows towards this direction. This will help to, trap that. Nutrients will be trapped, and then erosion will stop as well. So this is, a typical example. Next slide. Now, how do we minimize the environmental impact so that we ensure sustainable farming practices? In doing that, we have to look at precision fertilization. How do we apply a fertilizer? Do you just put that arbitrary? So now we have, you don't add fertilizer or chemical agent because you just want to, add it. You perform soil analysis or soil health indicator analysis, and based on that, you can make decision whether or not you need fertilizer to be added to your farm. And if that needs to be done, then you can use, the latest technology that we have, such as GPS guided. There are drones, but these are still not available in developing countries or in smallhold farmers where it's just being run by the family. They can't afford to use the GPS guided application of nutrient or fertilizer, or sort of monitor runoff, which is one of the things we do here as well. We monitor runoff when fertilizer is affected, is applied. How do we monitor their migration? So we look at that in runoff body, and then we can use that. We have a program that has been developed in-house, to monitor that runoff. Then we have targeted nutrient efficiency, where you apply fertilizer based on the different, sections of your farm. They may need different fertilizer, and all of this, again, I said, is guided by your soil health indicator. So the fertilizer is just not applied. Say, okay, NPK 30 or 20, 15, 15, and then you just spread that all over the farm. Some part of the farm may not need that. A typical example is in here, the farmers tend to add a lot of nitrogen fertilizer. So from 2 years ago when I joined, I found that they don't even need to add pea fertilizer. It's not needed because there's a lot of phosphorus fertilizer here. But before I developed this approach, they didn't know. Every year they were putting phosphorus. So that's going to cost the farmer money. And once there's increase in production costs, that is going to affect the output. It's going to affect the prices. But in the UK, you can't just put the price of food like that. It's being controlled by government, and that's why the government has got subsidy. So, but if you determine your soil health indicator year in, year out, some year you might find that you don't have to add anything. So with the P, what I'm trying to work out now for farmers is how often. They don't need it now, but what point do we get when they need it? So this information is what is very critical for the policymaker. So we can come up with something and say, for example, you have enough phosphorus for 3 years. So in the next 3 years, don't add phosphorus in your farm. And then that way, we would reduce the amount of phosphorus that is going to the water body that I have mentioned. That is a big problem now that is causing a lot of political, divide within the country. So you can use that and then strategize on where to use fertilizer and when not to use. And obviously, that would still improve the crop productivity and minimize the waste of fertilizer, and then cut the cost that you're putting in farm input. One other thing that I found that a lot of times this is what people ask is that, okay, if we go the natural way of doing stuff or a sustainable farming practice, how do we balance that with crop yield? Does it affect our yield or does it not affect our yield? So it's a fine balance. Either you produce a lot over a period of time, and you get to a point, it gets to plateau, and then it comes back, your soil is degraded. Or you produce in a sustainable way that it never runs out. You understand? So it's not making a big profit margin 1 year, and then 1 year you're running at loss. So that is not a sustainable way. What you want is produce your crop as a sustainable way. So what we find out from different kind of experiment, there's no big difference. Yes, using chemical pesticide may be a bit higher, but on the overall, if I was to compare it over 3 years or 5 years period, you would find that somebody who had gone on sustainable farming practice is better off than somebody who has been putting money to buy chemical herbicide, all of that, fertilizer, all of that into the farm. You find out, so the person doing chemical herbicide, all of that, in the first 2 years, they make more money. But once you go to the third year, then that match up with sustainability farm. If not, the sustainable farm hasn't gone up. And then by the fourth, fifth year, or 10 years down the line, you're going to see a decline if you're using all of that chemical inputs. Whereas this person, the production is still the same year in, year out, which is what you want if you're talking about sustainability. Then we look at organic farming. This is common. Now rich people want organic food. So if you go to the UK supermarket, Marks and Spencer, Tesco, Sainsbury's, Aldi, Lidl, you see that you have cashew nuts. This one says, you have banana, organic, non-organic. You find one is more expensive because people who have money, they want organic food. They want nutritious food. They can put any amount of money to buy. So for them, it's not a problem. So you're not, so by the time you put that cost versus just using chemical to produce over time, you find that someone who has gone organic farming is well off, over a period of time. And this is a common practice in the UK as well, and it cuts across vegetable and livestock, and they are all in the market. So this is a market, is there, it's just exploiting that value chain. Then we have waste reduction. You reduce the amount of crop that has been wasted and waste food. Instead of just throwing them, then you put that back. That way, you are ensuring carbon sequestration. You're putting carbon back into the soil, and which is good for improving soil organic matter and then the nutrient, in the soil. And then you could feed that into biogas or compost, and then just do anaerobic digestion. I have an experiment which I've done with that, and I can tell you there's a lot of improvement, versus when you use a chemical fertilizer. But because that data isn't published yet, I didn't put it here. But I'm happy to discuss about that, if it comes up in the section, question section. Next slide. So this is on this side, you can see drone there, applying precision, herbicide. You have the compost on this side, and then you have the biogas, organically produced vegetables, and all of this thing fits in. Like I said, in the UK, it's a big market. The organic fruit or vegetables market is very huge here. Next slide. So we have talked about a lot about all of this. What are the typical examples? Do we have examples around the globe? So I have taken a couple of examples as much as possible, just to give us an idea that this is working. We have seen results. In here, we have farms that we're monitoring. Some have been there. We have an anaerobic digester that has been running since 1970 something, and it's still running. We have farms that they haven't received fertilizer for the past 10 years, and they are producing. So next slide. So this is one typical example. We have the LEAF program in the UK, which is Resilient and Ready program. It's done in collaboration with the Cotiva Agri-Science. So they help to train and support future farmers so that they can adopt and promote sustainable farming practices. And this training is tailored. You go to them and say, I'm a small scale farmer, and this is what I do. They understand, they do a baseline study for your farm, and then they train you, they mentor you, and then so you can carry this on farm innovation so that you can then build. So if you check their website, which you can find here on the slide, their vision is global farming and food system that delivers climate positive action and builds resilience and support the health, diversity and enrichment of our food, farms, and the environment and society. Their mission is to inspire and enable more circular approaches to farming and food system. And this involves integrated, regenerative, vibrant, nature-based solutions that can drive productivity and prosperity among farmers, and also enriching our environment and possibly engaging the young people and the wider society. So a lot of young people have been trained on this program, which has recorded a huge success, in the UK. And then we are seeing the value of that as partners in the program. Next slide. This is another one which is called the Climate Farm Demo. The Climate Farm Demo, it cuts across 28 countries in EU, including UK. And the idea is they want to accelerate adaptation of climate smart farming practices and solutions by farmers and all actors of the climate smart agricultural knowledge and innovation system, which is called the ACAS. The view or the whole goal of this is adopting an agricultural production system to the climate and achieving a carbon neutral agricultural sector by 2050. That is also part of the EU climate strategy target. So if you check their website, they have a lot of thematic areas. So we do a lot of grassland management and forage and crops and then soil health. So this is where I fall. Everything I do is in this section. So there are a whole lot of them that potentially one can, read up more. And the link is just here if you want to read up more about them. And then, you gain more insight and see how you can build collaboration if they are looking. But at the moment, I think they are within these 28 European countries. But again, science has no border, Next slide. And then the UK Center for Ecology and Hydrology, this is another center, very excellent center. They've got a lot of farms, it's a center that's part of, the university, but they're independent. And then there's over 600 researchers that are working there. And all of them, what they're doing is finding solutions to all of the problems that our environment currently face and the one that the environment is likely to face in the next years to come. This, cut across climate change, pollution, biodiversity loss, basically, and they work a lot with researchers, and in trying to solve some of these problems. Then they encourage regenerative practices. They provide tools for land use management and planning, and evidence-based solution. And then they've been consulted a lot, just the way we are in Northern Ireland. A lot of policy that the UK government adopt comes from that center. Next. So this is a couple of cases. Again, I've tried to take just 3 each, just so I can keep within time. Next slide. Now we have the International Center for Agriculture Research in the dry areas. It's called ACADA. That, again, they promote sustainable farming in dry lands, and it cuts across 29 countries. And again, we're talking about climate smart practices like drought resistant crops, conservation agriculture, nature-based solution, or integrated livestock. You have the livestock in the farm, and then that way you control their grazing so that they don't graze freely in the farm. You can direct that maybe by petitioning the farms. One day they are here, while they're grazing, they put in their manure. It's a way of putting the carbon back, and then the next day you shift them to the other section of the farm. That has been found to be very effective. And the goal of this organization is that they want to improve soil health, water management, robust productivity, and enhance resilience to climate change. Again, they also work with farmers, especially women and youth. So that's what they're doing. So in a way, you are promoting this category of people. Whatever information they get from there, they then put that into global agricultural, policies that will strengthen food production and rural livelihoods, especially those that are vulnerable to climate extremes. So if you click the link, that will lead you to the site. You can lead up more and then see potential for collaboration as time arise, they might seek collaborations or building new teams across different parts of the world, and it's one thing that you could explore. Next slide. And then we have the project drawdown, and this one is based in basically what they're doing is they're using story to tell their solution. They come up with a solution which follows research, and they cut across different race, looking at people of different color, women, workers, all of us think in a different way, but the overall goal of the project is that they're encouraging neighborhood participation and a global solution for the dairy industry now. And all of their projects offer a real-time solution. So if you check their website, they say they are the leading guide to science-based climate solutions. That's all they do. They provide climate solutions that involve everybody, and the way they say it depends on who is involved, who is saying the story, and that's how they do it. And they just don't come and say, well, this is it. They say it in the form of a story. So it captures the intended audience for which the information is needed. Next slide. The watershed, that's based in Indiana in the US, and basically they are an NGO, and what they are doing is that they are protecting and improving the quality of water. This is all about water management now, in lakes and streams, in a particular region of Indiana in the US. And how do they do this? Basically, they help local farmers to promote sustainable agriculture, which comes, basically to water quality. And the idea is if you have a water quality and a sustainable use of water, that is going to promote soil health, which is one of their initiatives. And once that happens, they have things like the no-till farming, cover cropping, and nutrient management. And then they provide pre-technical assistance to farmer on ways you could reduce the cost of input in your farm, on alternate fundings for your farm if you're doing any of this, especially within the European Union as well. If a farmer is implementing any of this, there's a lot of support by the European Union that could support you if your farm is going to implement each of these, and they will help you, guide you, and implement that. In the Republic of Ireland, which is not, there's no border, physical border between Northern Ireland and Republic of Ireland. They even have money when you convert your farm to an agroforest, where you plant maybe crop or fruits. The government will pay you, per plant or whatever way. They have a way, to pay farmers, and then they come out and monitor each of these ones to make sure that it is successful, it doesn't fail. So that's why I said, in the short run, it may seem if you're using chemical pesticide and synthetic inputs in your farm, you may be making a lot of profit, but it's only over a matter of time that goes down. But if you're going to do any of these sustainable practices, you get a lot of support, and then that is sustainable over a long time period. Next slide. So, having said all of that, what are the challenges and what are the opportunities? So, if you want to start up any sustainable farming practice, like I've said, you need a baseline data. And the baseline data will mean all of those physical, chemical, biological data I spoke about earlier. You have to do all of them. So the initial cost is too much. So, and that's why, European Union and the like, even the African Union, I know that there was a time they called for a grant. So they are now looking for ways of how to support farmers in African countries to be able to look at that. I'm not particularly sure what is happening in Asia, but I know, they would have their own local arrangements there to take care of that initial costs, maybe through grant or seed money, that the government would don't, find a way of taking that. In Northern Ireland, for example, instead of the money you get as part of your carbon credit, they put it there. Or instead of some of the tax you pay to the government, if you put in any of these sustainable farming practice in place, the government will cut that tax for you, just to give you a bit of a relief over time. And so the huge costs, limited knowledge that we have, and then maybe training, market access, and then because at the moment it's all government driven or NGO, but farmers are looking at maybe a commercial way they can handle that. Because for example, one of the concerns the farmers have in Northern Ireland is we work for the government, and that government department we work for is the one that is regulating the farmers, is the one that is monitoring the farmers. So somehow some of the farmers don't trust us because they think if we have any information, we will tell the government agency that is paying us. And for that reason, some of them don't want to be part of that because they don't want us to know what is happening in the farm. But if they have an independent company that is providing that training and that support, they could go ahead and do. And that's what, as much as we can now, we're trying to make the farmers happy, give them some assurance, while we're trying to keep our paymasters, because they pay our salaries. We're trying to keep them happy as well. And while we come up with a sustainable way, of mitigating this challenge. There too, we have environmental as well. All of the environmental challenges that we follow, sustainable practice, you have to look for ways, to improve soil health, water quality, biodiversity protection. And then overall, once you do all of this, the overall idea is you want to boost the ability of your farm to be able to fight all of the environmental extreme or pesticide extreme, and then be resilient on their own. And then you have economic opportunities. Once you do all of that, like I told you, the organic market in the UK is huge. There's huge potential. It's more expensive. Even if you sell very few, because it's expensive, that serves as a makeup to any potential shortage. People are looking for eco-friendly product and everything. So all of that, there's a good market at the moment, and it's worth exploring. Plus all of the government bonuses that you get, will help to entice farmers to go into that. And then what is the overall outlook? Despite all of the obstacles that we have mentioned all through the presentation, one thing is very clear, is that the future of farming is a sustainable farming practice. If you want to be successful going forward with farming, then you have to embrace and adopt sustainable farming practices, and because that is just the direction that the world is going. And then there's a lot of global focus and support for that. There's a lot of collaboration. And then we're seeing a lot of grants as well, grants supporting work in sustainable farming practices. If you look at 10 years ago, there were fewer grants in soil and sustainable farming research than we have today. So it's something good. And if the bill, to the United Nations goes through, then the role of microbiologists and microorganisms within the climate change fight will be intensified. And then we would have a lot of other opportunities that will open within the field of sustainability and the role of microorganisms in that direction. Next slide. Yeah, so in conclusion, embracing a sustainable farming practice is vital for ensuring long-term food security and protecting the environment. Practical examples from the UK and across the globe have highlighted some real-world benefit and feasibility of implementing a sustainable farming practice. The next thing that we have to look at is achieving widespread adaptation, and that will require collaboration, knowledge sharing among farmers, researchers, policymakers, and consumers as well. And as a call of action, I would like to encourage all of us, to learn more and actively participate within our sphere in supporting sustainable farming practice where we find ourselves. I know, with AI now, there's a lot going on, but to be honest, one of the things that has been predicted, it is our job as microbiologists, as agriculture. This is the thing, is the only job that has been focused that it doesn't seem like in the near future AI is going to take our job. But the IT people, you know, that's the bonus for us. AI cannot take our job. Yes, it will take all of this, you know, the coding and all of that, fermentation, you put that. The biotech company I was working, by the time I was leaving, the guy who was working there was an India, you know, he's from Hyderabad, I think. So he left. But they were digitalizing everything. So AI does their fermentation. They can produce everything because it can monitor COD, monitor all of everything, every pH, temperature, all of that thing. But as a microbiologist, it's one job that there's no, at least in the next couple of years, AI is not going to take our job. So once we embrace this and begin to make microorganisms and microbiologists, you know, being felt, or, you know, we disseminate our importance, this will give us an edge, you know, moving forward, especially with the challenges and the prospect of that AI is coming with. So thank you very much for your time and the patience, you know, for listening to me for the past 1 hour now, I suppose. Thank you very much for the invite, Dr. Sharma.
Thank you so much, sir. And the session is open for discussion. If any questions are there, the participants can unmute themselves and ask the questions. If someone wants to ask any question, you can have that. Unmute yourself and you can ask the questions. I think so, sir. You explained so well that no one has any doubt about this topic. And the topic which you delivered, sustainable farming practices, cultivating a greener future, was very nice topic and up to mark. And is the today's day's need about the sustainable culture and the sustainable development, when we want to attain the 17 SDGs goal by 2030. Your scholarly deliberation has broadened the academic horizons of our participants by linking the concepts of sustainability you touched, the biodiversity conservation, the soil microbiome health, and climate resilience with practical agricultural strategies. So if anyone want to ask question, you can please go ahead. Sir has explained very nicely about the sustainable farming, which is not only about enhancing the productivity, but also about safeguarding natural resources, maintaining ecological balance, and ensuring food security for future generations. He highlighted innovative practices such as organic farming, water conservation technologies, crop diversification, no-till farming, integrated pest management, and soil fertility enhancement, and provided the participants a comprehensive understanding of the scientific as well as socio-economic dimensions of sustainability. His emphasis on the soil microbiome as a critical health indicator of soil has been very useful and has given a platform to our participants for a modern scientific perspective that connects microbiology with sustainable agriculture in a very meaningful way. And the knowledge and practical wisdom which you has enriched with the help of the global case studies and examples you presented, from ICRADA initiatives in dryland farming to leaf programs in the UK, have inspired participants to think that the conventional practices and to adapt context-specific innovations for the agricultural scenario, and moreover for the Indian agricultural scenario. We are truly honored, sir, to be benefited from your expertise, and we deeply appreciate the time and effort you devoted to this academic engagement. Despite your international commitments, your lecture is undoubtedly serving as a strong foundation for future academic collaborations, research initiatives, and for the advancements in the field of sustainable agriculture. On behalf of our
Thank you very much.
On behalf of our institution, Swami Shri Swarupanand Saraswati Mahavidyalaya and ATAL FDP team, I once again extend my sincere gratitude and look forward to continued association with you in the journey of promoting knowledge, innovation, and sustainability in agri-food systems.
Thank you very much, Dr. Sharma, and it's a pleasure to have you all. And open, if anyone needs to reach me, just get Dr. Sharma. She knows how to get me. And usually I'm quick to respond.
Okay, sir,
I'm open to any way I can help. Yes, I'm always happy to do
You
that.
responded very early, sir.
Yes, any way I can help. Yes, I'll be happy to
Yes.
to look at that and see how we can all contribute and share experience, collaborations, and knowledge and stuff like that. Thank you very much all for your patience and the time.
Rupali ma'am, you want to say something, please?
No, actually, I want to know, sir, elaborated that that there are some AI techniques that are getting used in precision farming and all those things. I just want just some elaboration on that.
I think, Dr. Sharma, that is HOD computer science, if I'm not making mistake.
Yes, she is from the computer science only.
Yes, I know. Yeah, so they are taking everybody's job. And so, now there are drones that can go in and do mapping. So here where I work, we have a geospatial department. So we put drone to go out and we do what is known as above ground. But remember when I started, I said we have below ground. So everything you see above ground with the crop, good yield, good fruit, is from the ground. If the ground is good, the up is good. So now we're using AI robotics to go out and collect data, especially because here is an island. So sometimes we have heavy storm, all of those environmental extremes. So we go collect that data. So that information is going to AI to predict future occurrence and how we can do that. One typical example just today, there was a call for a grant. I just finished putting a note. See that? So what we're doing here is a modeling. They want to be able to model crude oil spill.
Okay.
Because we have a ship dock here. So what they want, so they saw my thing in the press. So in the last couple of 3 weeks, I've been in the press here in the US, and tomorrow another one is going to be released in Germany as well, in one of my papers that I developed. So, which is based on biology. So what they want now is when those ships come, and if there's spill, how can we predict what is happening?
Okay.
So we know once they spill, microorganisms are going to respond. How they respond, I can predict. Dr. Sharma can do that, but maybe not computer people. So what is happening is when you have a spill, it goes on top of the water. Oxygen will not penetrate. If oxygen doesn't penetrate, that affects everything, all of the whole systems.
Yes.
Yeah.
Sir.
So now we depend on microorganisms, and through beta oxidation, they can attack that and convert the hydrocarbon CH3, and then they add oxygen from the air. And that makes
to the oxic world.
Yes, that makes it an aldehyde, and from aldehyde that becomes a carboxylic group, and then that can be breaking down. And once that is breaking down, that paves the way for fish to get oxygen, for zooplankton, phytoplankton. So in this model we're doing is an AI model that we're going to use, to sort of monitor that. What happened if there's a spill, in the event there's a spill. I mean, the model, they also want us to add, you see, when there's an emergency and the flight is forced to land on that water body, there might be spill. What is going to happen? So we're going to do all of that simulation. Yeah, this is the good part of computer. Microbiologists, we don't do that. I don't know what's going to happen with a flight emergency line, but apparently with computer, you can predict all of that and say, once it lands, this is what is going to happen, and all of that mathematical calculation, and you can work that out. So this is potential. But in terms of isolating DNA, running agarose gel, SDS page, all of that, AI cannot take that job. They still need us to do it.
Actually, AI needs knowledge, and knowledge, we will get knowledge from definitely from the life sciences.
Yes.
then and only then we can build this type of machines, this type of robots.
Yes.
So the knowledge is important, which will be provided by definitely the by life sciences faculties, life science scientists, and then we can perform these operations, algorithms. We can apply these algorithms and build such type of robots.
Yeah. So that's
It's
the
the
thing.
basic knowledge.
I thank the
That is
computer
not
for doing.
possible according to
Yeah.
Aliyu. He is the perfectionist as a microbiologist. He will never let it
I'm an
down.
advocate for microbiology.
Yes, yes.
Thank you,
It's
sir.
the fact that
We
you
go
rule
and
the
look for the
world.
data. And if you ask computer to do it, they say, give me data. But you need us to give the data. So you can't take my job. Give me my job and do your job.
Thank you so much, sir. Thank you.
All right. Thanks very much. Thank you,
So,
everyone.
Rupali ma'am, please ask Yogita ma'am to.
Yeah. Yes, ma'am, definitely. Okay, with this, we come to the conclusion of day 2 of our FDP. On behalf of organizing team, I, Rupali Kharche, sincerely thank both our esteemed speakers for their time and knowledge, our coordinator, Dr. Shamai Beg, for her constant support, and all participants for their active engagement. Now I formally express gratitude. I take the privilege of inviting Miss Yogita Lokhande, Assistant Professor Microbiology, to deliver the vote of thanks. Please, Yogita.
Thank you so much, Rupali ma'am. Now, as we conclude today's session, it's my privilege to propose vote of thanks on behalf of Swami Shri Swarupanand Saraswati Mahavidyalaya of second day AICTE ATAL FDP on technological innovation in agrotech and food processing. First and foremost, I extend my deepest gratitude to our esteemed speakers. We are highly grateful to Dr. Prakash M Halami sir for his enlightening talk on post-harvest technologies, biopreservation, and cold pasteurization. His insights on sustainable preservation techniques, biopreservation, biosensors, role of bacteriocins, and catalase application in cold pasteurization have greatly enriched our understandings. Thank you so much, sir. Our sincere thanks go to Professor Ibrahim Aliyu Dabai, sir, for his valuable session on sustainable farming practices, his global perspective on water management strategies, pest management methods for minimizing environmental impact, and case studies has offered participants fresh dimensions to reflect and adapt in real world agriculture. Thank you so much, sir. I also
Thank you very
wish
much.
our patterns, whose constant encouragement and guidance remain a source of inspiration for all of us. My heartfelt gratitude to our respected principal, Dr. Hansa Shukla ma'am, for her continuous support and motivation in making this program successful. A special word of appreciation to our coordinators, Dr. Shama Ibek ma'am, and our co-coordinator, Mrs. Rupali Kharche ma'am, and the entire organizing team who have worked tirelessly behind the scene to ensure smooth execution of today's session. Last but not the least, I thank all the participants for their active involvement, meaningful discussion, and thoughtful interaction that made today's session truly engaging and fruitful. Before we conclude, may I kindly remind all the participants to fill the feedback form, as it is also serve as attendance, and to join tomorrow's session on the time for another enriching day of learning. Thank you so much.
Thank you.
Thank you, Yogita. So the today's session is over. Thank you, Aliyu sir, and thank you, Rupali ma'am, and all the participants, because it's you only who is making this FDP a valuable one. Thank you so much to all of you.
Thank you very much.
Thank you.
Good night, everyone.
Okay. Bye-bye.